Probiotics and Prebiotics from Bina Nusantara University, Indonesia. Haruhisa Hirata, PhD. The academics and practitioners of probiotic from Japan. Patricia Conway, BSc, MSc, PhD, adjunct professor of gastrointestinal, metagenomics, and metabolomics, functional foods, and foods, probiotic and prebiotics from Australia. Dr. Ee Seong Thi, the president of Nutrition Society of Malaysia. Distinguished participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Good morning and may peace be upon all of us. We would like to extend warmest welcome to all of you. It is great pleasure for Indonesian Food Drug Authority to host this virtual seminar and exhibition with the topic of research and potential development of prebiotic products for health and regulatory study of probiotic in other countries. The objective of seminar is to share information related to research and the regulation of probiotic in the USA, Australia, Finland, Japan, Indonesia, and ASEAN. It is our hope that this knowledge sharing program will be beneficial for us to increase knowledge and understanding in developing health supplement products containing probiotics and as a reference for the Indonesian FDA to make recommendations to conduct communication, information, and education regarding the correct use of health supplements containing probiotic for the community. Excellencies, ladies and gentlemen, please allow me to read briefly on the today's program. For opening session, we will hear remarks by Her Excellency, Dr. Penny Kelukito, MCP, the chairperson of the Indonesian FDA. Next, virtual group photo session. Excellency, participants, ladies and gentlemen, without further ado, now we cordially invite Her Excellency, Dr. Penny Kelukito, MCP, the chairperson of the Indonesian FDA, to convey her message and will kindly officially open this virtual meeting. Please welcome Her Excellency, Dr. Penny Kelukito, MCP.
Distinguished speakers, Mr. Haruhisa Hirata, PhD, from Wakamoto Pharmaceutical, Japan, Professor Ingrid Suryanti Suryono, MSc, PhD, President of the Indonesian Scientific Society for Probiotic and Prebiotics from Binus University of Indonesia, Professor Jan Peter van Peekeren, University of Wisconsin-Madison, USA, Ee Seong Thi, BSc, MSc, PhD, President of Nutrition Society from Malaysia, and Patricia Conway, MSc, PhD from University of South Wales, Australia, Professor Seppo Salminen, MSc, PhD, President of the International Scientific Association for Probiotics and Prebiotics from Finland. And then also yang kami hormati Bapak Ibu sekalian panelis dari berbagai perguruan tinggi, Ikatan Dokter Indonesia, dan juga dari industri yang hadir dalam kesempatan ini, para partisipan dari asosiasi, baik itu pelaku usaha, asosiasi profesi, dan akademisi, dan tentunya juga para jajaran Badan POM serta Bapak Ibu sekalian. Assalamualaikum warahmatullahi wabarakatuh. Good morning and good evening. First of all, I extend my warmly welcome to all participants of the International Scientific Seminar on Probiotic titled Research and Potential Development of Probiotic Products for Health and Regulatory Study for Probiotics. Indonesian Food and Drug Authority, Indonesia FDA, is pleased to convene this webinar to discuss. This is webinar seminar to discuss. First is the research and potential development of probiotic. So clinical study for probiotic product claims, especially for its benefit for dietary supplement. And third is for marketing regulatory overview. Let me also convey our gratitude and appreciation to all speakers that coming from all over the world, the USA, Finland, Australia, Malaysia, Japan, and also from Indonesia. We are grateful for their valuable, competent knowledge and experience for probiotic, which I believe coming from various points of background of view also to enrich our in-depth knowledge about probiotics and their value for human health. Distinguished participants, ladies and gentlemen, probiotic has scientifically approved benefit to the human gut and improve overall health. And probiotic made our body maintain a healthy community of microorganisms and return our body microorganism to a healthy condition after being disturbed, for example, and influence also our overall body's immune response. Thanks to the development of science and innovation in technology, probiotics can be edible as also food products. Among the increasing research and development on probiotic and their potential health benefits, product safety, and also the effect and the claims on label and advertisement are the most important aspect that we should consider. So in this regard, we need regulation, technical guidance, such as based on clinical study, and also scientific literature as reference to comply with the requirement on the safety and the right communication, a proper communication on the beneficial effect, the claim of a probiotic products. We understand that every country has its own policy on probiotic regulation, which depends on the authority, intended use, and marketing approval. For example, in the United States, probiotics are regulated under US FDA surveillance. Probiotic may be regulated like fruit ingredient, dietary supplement, or for a drug, depending on the various intended use. Meanwhile, in Europe, the European Food Safety Authority doesn't approve any claims of health benefit for products containing probiotic, except for the claim for gastrointestinal health. For Indonesia, the Indonesian FDA has issued our own regulation number 17, just the year 2021, just a year ago, on guidelines for the assessment of health supplement containing probiotic. We are in the process to complement our new regulation on probiotic supplement with technical guidance and required scientific evidence for validating probiotic health supplement for each intended use for marketing authorization. Distinguished participants, ladies and gentlemen, this seminar offers an excellent opportunity to gain new insight about probiotic, especially on research on new probiotic strains. From this forum, we expect also to acquire scientific feedback for the Indonesian FDA in our process in developing technical guidance for health supplement product containing probiotic. So I wish you all fruitful discussion and share knowledge on current research, potential development, and also regulation related to probiotic product for human health. In conclusion, I declare this virtual seminar officially open. Thank you very much.
Thank you, Madam Chairperson, for your valuable remark and has officially opened this important meeting. We cordially invite all of participants to turn on your video for photo session. Please kindly use the given virtual background, stay on the video and keep smiling as the host would need a few minutes to capture all the pictures screen by screen. Okay, please turn on the virtual background, turn on the camera. The host will capture screen by screen. Excellency, ladies and gentlemen, we cordially invite Her Excellency, excuse me. Cordially invite Her Excellency Ibu Reri Indriani, Deputy Chairperson for Traditional Medicine Health, Supplement and Cosmetic Control, Indonesian FDA, kindly to visit the product exhibition. Please welcome Her Excellency Ibu Reri Indriani.
Iya.
We will hear the presentation from Deputy Chairperson for Traditional Medicine, Health Supplement and Cosmetic Control, the Indonesian FDA, Ibu Reri Indriani. The title of the presentation is Overview of Probiotic Regulation in Indonesia. Please welcome distinguished Ibu Reri Indriani.
Thank you, Bu Pulan. I hope that everybody who is in Zoom, also in this aula, can hear my voice clearly. Distinguished ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Good morning. First of all, I would like to thank you for all speakers and participants in this scientific international seminar on probiotics. In this valuable event, I would like to present about the overview of probiotic regulation in Indonesia. My presentation will consist of 4 major topics. First, I would like to introduce you to Indonesia FDA regulation on probiotics. And second, about the probiotic product category. And the third is about the probiotic as health supplements. Because under BPOM regulations, category of the food supplement have 3 categories: as the medicines, as health supplement, and also as food. And the fourth is about the potential development of probiotic as health supplement. Next slide, please. Yeah. First of all, I would like to introduce you, maybe some of you that not familiar with our organizations, about the Indonesia organization and how Indonesia FDA regulates probiotic according to Indonesian law. Yeah. Indonesia Food and Drug Authority, or we call it Indonesia FDA, is a non-ministerial government agency that carries out government affairs in the field of drug and food control. The Indonesia FDA has the responsibility in surveillance and controlling pharmaceuticals, biological products and vaccines, narcotic and psychotropics, traditional medicines, cosmetics, health supplements, processed food, and hazardous substances. As we seen here, this chart describes the organization structures of the. Ini mohon zoom-nya jangan menutupi slide saya. Di sini, nah ya. As we have seen here in this chart, the chart describes the organizational structure of the Indonesian FDA. Probiotic is regulated as food product or as health supplement products. When a product containing probiotics and it has beneficial claim intended use to health supplement, then it should be under Deputy for Traditional Medicines, Health Supplement and Cosmetic Control. Next slide, please. How we define probiotic product category. In line with the International Scientific Association Probiotics and Prebiotics definition, the definition of probiotic in Indonesia define it as live microorganism that when administered in adequate amounts confer a health benefit on the most. In Indonesia, the probiotics may determine into three categories. First, as medicines. Secondly, as a health supplements that we are talking now about. And third, as the processed food category. Yeah. In this seminar, we look forward the discussion would be catch on probiotic as health supplement. Next slide, please. In line with the Perbadan number 17, 2021, based on this flow chart, probiotics in the category of medicines and health supplement are formulated in pharmaceutical dosage form, yeah, such as capsule, etc., or tablet, etc. But the difference in the terms of claims. Medicine, probiotic as medicine has claim for treatment to cure or curing the disease, while health supplement has more light or general claims. Yeah, this definition both according to the Indonesian regulation, which mentioned or stated in the Badan POM number 17, 2021. Probiotic in processed food category are formulated as food or drink, and the things to be considered such as serving size, sensory characteristic as food and drink, such as quantity, taste, and also in line with the standard for food category and how to consume, serve as food and drink. In this presentation, we focus on probiotic as health supplement, that this product under the Deputy for Traditional Medicines, Health Supplement and Cosmetic Control. Next slide, please. We are now talking about probiotic as health supplement. In 2021, Indonesia FDA has permitted and published the regulation number 70 concerning guidelines for assessing health supplement products containing probiotics. This regulation aims to provide guidelines for evaluating health supplements product containing probiotic as a reference for our internal evaluators and also for industries in submitting their product to be registered in Indonesia. About the general principle in probiotics that need to be considered, first that the living microorganism should be in adequate amounts that can provide health benefit for human and could be single or combination microorganism as a product and provide benefit for balancing of the digestive tract microbiota. Microorganisms as probiotics should comply with some criteria and evidence, such as probiotic term with the right amount of probiotics, has the ability of probiotic to proliferate in the digestive tract or in colon, environmental influences and local life habits, genus, species, and strain characterization, antibiotic resistant genes, and scientific evidence to support safety and claim benefit also needed. About the requirement of product registration, the registration requirement for health supplements containing probiotics are related supporting documents such as safety data and benefit document, and of course, quality product document. Distinguished ladies and gentlemen, Bapak Ibu sekalian, in line with the Perbadan POM number 17, 2021, in this regulation, we have the flow chart that explain, as you can see in the screen, that the flow chart that explain the principle of evaluating supplement products containing probiotics according to the FAO and WHO guideline. Based on the flow chart, a product containing probiotic could be evaluated and categorized as health supplement with documents submitted for identification as the FAO and WHO guideline. And the assessment steps of the evaluation including or involve first the probiotic strain identification. This is the very most important steps. Yeah, the identification of the strain of the probiotic used in the health supplement product. Secondly, also very important is the strain characterization. Yeah, safety review and efficacy study is a must. Yeah, all these four components cannot be separated. This is an integrated data to support the safety, the efficacy, and also the quality of the supplement product that contain probiotic. Next slide, please. Now, in line, according to the previous flow chart, yeah, the assessment step for evaluating health supplement products containing product consists of first probiotic identification. Is phenotyping or genotypic method to determine the genus, the species, and deposit strength in international cultural collection. Yeah, usually the international cultural collection have their database. It's a very complete and comprehensive and up-to-date database. Secondly, strain characterization by in vitro and vivo tests. And the third is safety review by 3 data from in vitro, in vivo tests, and phase 1 clinical trials. And the next step is the efficacy study data, including phase 2 clinical RCT randomized control trial or other appropriate design with sample size and primary outcome appropriate to determine if the strain of the product is efficacious. Yeah. In RCT to confirm, or in RCT to confirm that the result of phase 2 clinical trials. And also phase 3 clinical trial effectiveness trial is appropriate to compare probiotic with standard treatment or specific condition. This is in specific condition. Next slide, please. In addition to safety and benefit data, there are also requirements for quality aspect of health supplement product containing probiotic, such as the sources of probiotic, product specification test, microorganism contamination, and other quality document can refer to health supplement quality requirement regulation. This 4 document is necessarily needed by our evaluator. Bapak Ibu sekalian, distinguished ladies and gentlemen, to point out of health supplement claim evaluation, especially containing probiotic, it should be fulfill the 4 categories that the claim benefits are specific and supported by scientific evidence, and there are minimal amounts of probiotic which provide benefit. Yeah, and also duration of use according to supporting data. Yeah. Next slide, please. Now, about the labeling of the probiotic, that the health supplement containing probiotic label should include some information such as content, which in genus, species, strain designation, minimum number of variable bacteria at end of shelf life. This is important, yeah, at the step of end of shelf life. Instruction in storing condition, yeah, because this is to prevent that there is any destruction related to improper storage condition. Yeah, the product produced by the manufacturer complies the safety, efficacy, and quality control, but the community, the consumer, do not store it under the proper storage condition. Yeah, it's very fitty. Okay, we go back to the slide. So, Bapak Ibu, distinguished ladies and gentlemen, at the end of this my presentation, I would like to share about the potential development of probiotic as health supplement. As we can see in this pie charts, that according to the product data registered in Indonesia, it is dominated by imported product, which around 95%, yeah, or almost 96% product. While the locally produced in Indonesia or under license are only 2.70%. Most of the product has been approved with beneficial claim for maintaining health gastrointestinal. Yeah. This data shows that there are still many chances for industry in Indonesia to develop manufacturing probiotic products as health supplements. And of course, it should be complied with the requirement for approval as health supplement as well. One of the objectives Badan POM to conduct this international scientific seminar is to share the regulation to overview others FDA regulation in order to get benefit what might be suitable for Indonesia regulation in order to back up the Indonesian manufacturers in developing more probiotics for Indonesia community. Next slide, please. Health supplement products defined as a product that intended use to nutritional needs, maintains, enhance, or improve health function, has nutritional value or psychological effect that could be contains one or more ingredients in the form of vitamins, minerals, amino acids, and other non-herbal ingredients which can be combined with herbals. When products containing probiotic combined with other active ingredients and it intended use to be health supplement, there are many challenges on providing scientific data to meet with the requirement of safety, quality, and beneficial claim. So in this seminar, as I have mentioned before, we have to talk more about how to get best way to cover up the obstacle, especially on providing scientific data such as clinical data that support the beneficial claim as health supplements. This is still the challenge that the industry have, and our internal evaluator is try do the best in order the manufacturer, the industry to meet or to fulfill the Indonesia FDA regulation. Allow me to end my presentation by summarizing that in Indonesia, probiotic could be categorized as health supplement product as long as it comply with the requirement on safety, efficacy, and quality, and its definition as health supplement as mentioned in the regulation or Perka Badan POM number 70 2020 concerning the guideline for assessing health supplement product containing probiotics. As we have all know, since all the manufacturers, all the importers, and all stakeholders involved in the preparing this regulation, that the guideline about probiotics safety and efficacy refer to the WHO guideline. And the beneficial claim of product containing probiotic should be supported by scientific data, such as clinical trial study. It is very important to ensure that the product contains probiotic provide the health benefit claim as stated on the labeling. Therefore, clinical trial is needed to support the claim, and it has been regulated in Indonesia FDA regulation as health supplement containing probiotic as health supplement. Of course, I hope that all participants could gain knowledge and information about probiotics regulation in other countries. So this benchmarking would give another perspective about on how to regulate the probiotics as a product, especially if there are disclaimer on labeling. And also it is potential development as a product with benefit claim needed or supported with any scientific data. Yeah. During this seminar, all information and discussion would be useful for both Indonesia FDA and industry. I do hope that all information and discussion would be very useful for us in order to gain new insight on research and development on probiotics, especially in conducting registration approval as health supplement in Indonesia. Last but not least, that pentahelix cross-sector collaboration among government, business, academics, community, and media is very important and is needed in the development of broader probiotics claim, especially in additional to digestive health claim. Badan POM, as the part of the government, cannot be a single player. We need all the commitment from other stakeholders. Yeah, since so far, probiotics claim that approved by Indonesia FDA limit only to for health digestive. Yeah, we need to broaden the claim so that the consumer, the community get broader benefit from the probiotic, particularly during this COVID-19 pandemic. I think this is the end of my presentation. Thank you very much for listening, and I hope that you have all a very good all day. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Madam Deputy for Traditional Medicine, Health Supplement and Cosmetic Control, Indonesian FDA, for your valuable presentation. Besides seminar, there is exhibition that is located the right of this building. Please kindly, Ibu Reri Indriana, visit the product exhibition booth. Now we are going to the next session. For the first webinar, the topic is Regulation Overview, Research and Development of Probiotic Product in USA and Japan. With the speakers are Professor Jan-Peter van Pekkeren and Haruhisa Hirata, PhD. The panelists are Ibu Rahmi Setiorini, Dr. Rina Agustina, MSc, PhD, and Dr. Rini Listiana, MM. This session will be facilitated by the moderator, Ms. Wiwin. Over to you.
Thank you very much, Ibu Pulan. Good morning, distinguished speakers, ladies and gentlemen. Welcome to the International Scientific Seminar and Product Exhibition on Probiotic, hosted by Indonesian FDA. I'm Wiwin. I will be the moderator for the discussion session. Today, probiotic is gaining more popularity and attention due to its wide potential benefit. However, research and development of new probiotic is a lengthy and costly process. So, how is the future of probiotics in pharmaceutical industry? We are gathered today to discuss and share insight about the research and development of probiotic for health. In this first session, we will hear about the regulatory overview, research and development of probiotic in the USA and Japan. In this session, we have two speakers. First, Dr. Jan-Peter van Pekkeren from the University of Wisconsin, USA. Good morning from Jakarta. Dr. Van Pickeren, and good evening in the USA.
Hi, good morning. How are you? Here, JP van Pickeren.
We are very good, thank you. Thank you for taking the time to join with us today, Dr. Van Pickeren. And
very welcome.
And the second speaker is Mr. Haruhisa Hirata, PhD from Wakamoto Pharmaceutical, Japan. Good morning, Dr. Hirata. Very nice to meet you today. How are you?
Selamat pagi. おはようございます。若松製薬の平田です。
Thank you very much. And also, in this first session, we have 3 panelists. First, Ibu Rahmi Setiorini. She is the Director of Standardization of Traditional Medicine, Health Supplement, and Cosmetic, Indonesian FDA. Very good morning, Ibu Ririn. And the second panelist is Dr. Rina Agustina. She is the Head of Postdoctoral Study, Faculty of Medicine, University of Indonesia. Good morning, Dr. Rina.
Good morning. Thank you for invitation.
Yes, thank you very much for taking the time to join with us today. And the third panelist is Dr. Rini Listiana, Group Manager of Business Development, PT Labe Laboratories. Good morning, Dr. Rini. Thank you very much for joining us today. You're welcome. Okay, moving on to our main agenda is the presentation. Our first topic is regulatory overview, research and development of probiotic product in the USA. Before we listen to Dr. Van Pickeren, I will give you brief information about him. Dr. Van Pickeren is an associate professor at the Department of Food Science, chair of the Food Science Graduate Program, and the executive member of the Food Research Institute at the University of Wisconsin-Madison. He received a Bachelor of Science in Biotechnology from the Noordelijk Hogeschool Leeuwarden and a Master of Science in Biology from the Leiden University, both in the Netherlands. He joined the laboratory of Dr. Paul O'Toole at the University of College Cork, Ireland, where he completed his PhD training in microbiology. During his postdoctoral training, Dr. Van Pickeren developed Listeria monocytogenesis as a DNA delivery vehicle at the Cork Cancer Research Centre, Ireland. After completing his second postdoctoral training under Dr. Robert Britton at the Michigan State University, he started his own laboratory in 2013. Dr. Van Pickeren developed various genome editing tools for the use in undomesticated lactobacilli, which are essential to pursue his long-term research goals. Studies in the Van Pickeren laboratory focus mostly on gut symbiont species, Limosillatobacillus reuteri, until recently known as Lactobacillus reuteri. His research team aimed to unravel the molecular mechanism by which the gut symbiont interact with members of the microbiome and the mammalian host. Knowledge gained from these studies are leveraged toward the development of the next generation probiotic for the use in human medicine. Without further ado, I invite Dr. Van Pickeren to present his speech. Please, Dr. Van Pickeren, you have 30 minutes. The time is yours. Thank you.
Thank you very much for the introduction, and I am very grateful to be here, and I'm looking forward to sharing some of our work and to the discussion afterwards. So I'm going to give a presentation where I give a 20,000 view of what probiotics are, and some of that information we already have seen in some of the introduction. And after that, in the second part of my talk, I will talk about some of the research that we are conducting in my laboratory. And specifically, I've chosen to highlight one story that, that focuses on the mechanism that contributes to the ability of a select probiotic strain to thrive in the gut ecosystem. So I will highlight that, and that work is really fresh, and it appears later this week in the issue of Cell Host and Microbe. All right, so before I get going here, my university wants me to put this forward, this disclosure that I am the founder and owner of a consulting company. But in this presentation here today, I have the hat on of a professor of UW Medicine, so nothing in this talk is actually linked to the consulting company. All right, so having that out of the way, we already have seen the definition of probiotics, live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. And this has been put forward in 2014 by a panel of experts from the ISAP, and later Professor Salminen will, I'm sure we'll talk more about the work of the ISAP. When I look at a definition, its definitions are great. They give you a bit of insight, but at the same time, a lot of questions appear. For example, what about dead organisms? Well, dead organisms can certainly have a health benefit. They are not fall under the umbrella of probiotics. There is actually a different term has been developed for that. It's called postbiotics, but dead microorganisms in itself are not considered probiotics. It creates a bit of a gray area, though. The previous speaker highlighted shelf life being important, and if you think about probiotics sitting on the shelf, eventually you end up with a mixture of dead microbes and live microbes in your product. At the same time, though, it is the live microbes in there that are considered the probiotics. A bit of a delay here, I see in my slide transition. Which microorganisms? Most commonly known are, of course, the lactobacilli and bifidobacteria. But yeast, there is actually Boulardii, is actually Saccharomyces boulardii is actually a probiotic shown in clinical trials to ameliorate some diarrhea. Bacillus has been put forward as a particular probiotic, and actually one strain of E. coli Nissel, is also a well-established probiotic. So there is actually quite a spectrum of species that can fall under the umbrella of probiotics. We think of the aspect administered how? Well, mostly it is administered through ingestion, but there are also topical creams available for probiotics. And the ingestion can either be through a pill, or it can actually be part of a drink, or it can be in foods to which these strains have been supplemented. It is listed, it has to be administered in adequate amounts. Honestly, I think this is a very gray area, an uncertain area. Of course, you need to have enough microbes to have the benefit, but I think that currently this definition gives the idea that we know what the adequate amount is, and the reality is we do not know what the adequate amount is. Most clinical trials stick with a fixed amount that is typically around 10 to the 9th, and that is administered once or sometimes multiple times a day. But to my knowledge, no clinical studies have been performed really looking at the titration and determining what actually is the adequate amount. And that for sure will differ per strain and per application too. And then which host? Well, here we are talking mostly about humans, but probiotics have also been developed for use in agriculture, for pets, dogs and cats, and even for fish. So there's actually probiotics for any animal basically on the planet. Most probiotics have been put forward, and it's a huge market, as we all here are aware. It's a multi-billion-dollar market. I got this actually also again from ISAP, and if you're, I highly recommend everyone to have a look at their website. It's very informative. There's lots of information there, and this basically recaps what a probiotic is, what I just have talked about. But I just briefly want to focus here what probiotics, at least under the definition and for the ISAP, what are not considered probiotics. So fermented foods in itself are not considered probiotics. Yes, they contain live cultures. Yes, it has been established that actually fermented foods are considered healthy, but because this actually consists of an undefined consortium of microbes, this is not considered a probiotic. Another example of undefined consortia of microbes here would be members that are present in a fecal microbial transplant. So patients that are suffering from recurrent Clostridium difficile infection, where antibiotic treatment is not helping anymore, yet the infection continues, a cure that actually has a cure rate of over 90% is a fecal microbial transplant. And as its name suggests, a fecal transplant is transferred to the patient, and the cure rate is over 90%. Because clearly health promoting, right? It does not get more health promoting than that. However, we still are working with an undefined community here, which is unknown, and it is unknown which combination of microbes or which select microbes that are part of that community contribute to the health promoting effect. So therefore, at least these are not considered to be probiotics. At the moment, the FDA has not approved any probiotic to cure a particular disease, so there is no health claim for that. Instead, probiotics are regulated by the FDA as dietary supplements, and the claims that are listed with probiotic products and the strains that are in products are typically structure function claims. And what this basically means is an example here that this product contains bacteria that supports healthy digestion. So it's a very general statement, or promotes bone health as another general statement. But these structure function claims cannot contain language that links it directly to a disease. So it cannot say reduces intestinal inflammation or reduces cholesterol levels, because that actually is linked to a particular disease. So these are typically the structure function benefits there. If we think about potential probiotic effectors, there's actually quite a few, and I will not go over this slide here, but there's actually quite a list that you see here. For example, the production of short-chain fatty acids, lactic acid or acetic acid that can play a role directly modulating the host immune system. Production of bacteriocins, of course, which are antimicrobial molecules that can clearly have an effect in eradicating pathogens. And but all of these typically have been determined in preclinical models at most, right? A lot of that is also in vitro or in tissue culture, and several of these have been demonstrated an effect in preclinical models, i.e., mostly mice models actually. But evidence in humans is lacking. And here specifically, when I think of evidence, and I like mechanistic work, and when I think of mechanism, I here think about having a gene knockout, for example, and test for function. And you can already see the problem there with ingestion of a GMO, because if you knock out a gene, it typically is considered a GMO in testing and performing these studies. At the same time, though, there is actually a long list of clinical trials conducted pertaining probiotics or that contain probiotics and that are effective in humans. But the flip side of the coin is that the mechanism is typically unknown. And it is beyond the scope, really, and I don't have enough time to go through all these different clinical trials, but I actually guide you to a website that I've listed here that is generally accessible. And this website nicely has done that job already of collecting data of all different clinical trials conducted for different disease states. And the website subsequently provides you information with a so-called recommendation level, with a recommendation level of 1, which is the highest level of recommendation, where these studies are backed up by solid clinical trials, solidly designed clinical trials, for example. So, for example, if you were to click on adult health here, you go here, and if you can expand on one of these brand names here, Align, you can see here that the application is IBS, irritable bowel syndrome. It has the highest level of recommendation, and you have some references here. And I just encourage you to have a look at this website and to get a sense of actually the application of probiotics in the different clinical trials. Here are 60 products listed, and these can either be individual products or strain combinations, really. But all of these strains listed here and that are embedded are strains that have the so-called GRAS status. So they are generally recognized as safe. But as I said, it is unknown mechanism, and that actually is the little bridge to the second part of my talk, I should say, which is some of the work that we're doing in my lab. And we are really enthused and driven to understand mechanism, and our overarching goal is really to leverage the knowledge that we gain from the mechanism of how a probiotic interacts with its host and how it thrives in the host, to leverage that towards the development of microbial therapeutics. Now, to understand mechanism, we work with, as already nicely introduced in the introduction, we work with a gut symbiont species called L. reuteri, Limosi Lactobacillus reuteri, until recently known as Lactobacillus reuteri. And this is a species that is commonly found in the gut of a large number of vertebrates, including in humans. And for the last 10 years or so, I invested heavily to develop genome editing tools. Genome editing is not trivial in many lactobacilli, certainly not in bifidobacteria. And especially if you're working with strains that we call wild isolates, so strains that have been recently isolated, for example, from the human gut, these are typically resilient to genome editing and genome modifications. And it was just a bit of inspiration, a lot of perspiration to develop these tools. And we now have a really nice platform developed that puts us in a position to engineer L. reuteri, but also other lactobacilli. And I will not go into the details of these tools here, but all you need to know for now is that we can either make SNPs in the chromosome, we can make large insertions, large deletions, and we also have developed CRISPR-Cas genome editing in L. reuteri and other lactobacilli. So we really have a carte blanche for our ability to engineer a range of lactobacilli. And being able to engineer lactobacilli is, of course, key if you want to understand mechanism, because you want to be in a position to inactivate a gene to understand its function and to link genotype with phenotype. And as I already alluded briefly to, my lab is interested to understand gut fitness. What is it about select strains that allow it to thrive in the gut ecosystem? What are the underlying mechanisms? What are some of the mechanisms that contribute to the ability of a probiotic to interact with its host? And ultimately, we leverage that knowledge towards the development of microbial therapeutics. And I'm not going to touch on that part here. This is beyond the scope. But I want to actually focus on the aspect of gut fitness and tell you very briefly a story here that we recently developed. So if we really go back actually to the 80s and 90s, at least in the US, where probiotics were starting to show up more and more in literature, and think about the selection criteria that historically had been applied for probiotics, that often was a matter of adding some bile acids and adding hydrochloric acid. And the first checkbox here is that, okay, the bacteria, my lactobacillus, survives exposure to these acids. So, you know, it must do well here to survive gastrointestinal transit. And thankfully, we have moved away from that fairly quickly and try to understand the ability of an organism to thrive in the gut. You can fairly easily do that by passing it through the gut of a mammal, and now more and more also humans are used as test subjects, so to speak, to understand how an organism can thrive in the gut. And it is really as simple as taking in a probiotic and see what comes out and what the level of survival there is, if you really look at the basal level and really just want to get a gist if they survive. But in doing so, you are actually just looking at a single strain typically that is being ingested. And with that experimental approach, you are actually ignoring the trillions of microbes that inhabit our gut ecosystem. And these microbes are competing also for nutrients, and these microbes are also competing for niches, and there may actually be direct interactions that can be happening between the microbes and the probiotics. So if we think about selection of probiotics, and in an ideal world, you really want to understand mechanisms that can help us towards better selection of probiotics in the future. At least that is my 2 cents on that. And, you know, what I just have explained on the interactions that may happen, that is really on, you know, on a 10,000 feet view. But if we put our microbial ecology goggles on, so to speak, and if we think about the different types of microbe interactions, there is actually quite a few, as is listed here. And I will not go through these, but it just, you know, paints the picture of the complexity of the different types of interactions with the different microbes living there, the different species, the different strains, etc. So our goal really is to understand the mechanisms that select probiotics have evolved to compete with other microbes in the gut ecosystem. So for this work, this study started with work from Jens Walter. He's a phenomenal microbial ecologist who worked actually for quite a bit of his career on L. reuteri. And in one of the studies that he conducted was, he asked the question what the fitness was of select strains if you made a mixture of select strains and you put them through a mouse, put it through the GI tract of a mouse, and you ask the question, okay, who is being recovered at which levels? Okay, simple as that. And if you look at the picture at the top left here, we see that one strain in particular, strain R2LC, is being recovered at very high levels, whereas another strain, strain 100-23, is being recovered at log difference levels, okay? And you need to know that strain R2LC is a probiotic. It has been safely consumed by humans, and it is a strain that has been shown to ameliorate colitis in preclinical models. And we also have done some work with regards to understanding a gene cluster which modulates the immune system that this strain modulates. But I will just to mention that briefly. So this is in mice, but Jens went on and actually had tested, again, different mixtures of strains, including this strain R2LC. And one of the subjects that it was passed through was a human. And again, here, he saw that in humans, this strain R2LC is recovered at very high levels. So in other words, you know, depending or independent typically here of this mixture, we see that compared to most strains, strain R2LC is very fit in the gut ecosystem. And we wanted to know why, and that was our goal. So the work has been conducted by a graduate student in the lab. He graduated since, he's now a postdoc at UCLA, and Dr. Erskam discovered that this particular strain is a pigmented strain, so it is bright orange color, as you can see here. And we discovered by comparative genomics that this strain, or by genomics, I should say, that this strain codes 2 large gene clusters, which we predict code for secondary metabolites. And given the role that select metabolites can have in terms of antimicrobial activity, we asked the question, well, does one of these clusters contribute to the ability to outcompete strain 100-23 in the gut? And we set up a really simple experiment. We first, which was not so simple, but we managed, we used our genetic tools to inactivate this top cluster, this fun cluster, functionally unknown. And we also made a derivative in which we inactivated this so-called PKS cluster. And what we now had done is that we mixed strain 100-23. So again, I'll refer again to Jens's work here that was greatly outcompeted here by strain R2LC. And we just mixed those 2 strains together. We grew them up and we put them on a plate. And we basically looked which color colony we recovered, because we can distinguish between R2LC and 100-23 based on their color. R2LC is pigmented, 100-23 is opaque. And we see that R2LC on its own is, of course, pigmented. If we inactivate this fun cluster, we only recover pigmented colonies, which means that R2LC is outcompeting this still 100-23, even if we inactivate this fun cluster. But what we now see very excitingly, if we inactivate the PKS cluster, we now actually recover white colonies, these white speckles here, which is strain 100-23. So in this example here, R2LC has lost the competitive advantage. If we look in a bit more detail here, we see that this gene cluster is responsible for very rapid killing of strain 100-23. So we repeated the experiment whereby we grew both cultures in a single tube. And we measured strain 100-23 levels by plate counts over time. And we see after 6 hours co-incubation, we have this rapid decline here in the colony forming units, indicative of killing. And this killing is specific by PKS, the PKS cluster, because if we inactivate the PKS cluster, what we see, is that now actually the 100-23 levels continue to grow at similar levels compared to 100-23 on its own. So clearly the PKS cluster is responsible for killing of 100-23. And this now already gives us a bit of insight to the work from Jens. We then went to germ-free mice, where we repeated the experiment whereby we co-incubated this PKS-producing strain R12C, with 100-23. And I will not go through everything here, but just in the cecum as the example, we see here that strain 100-23 outcompetes, strain R12C outcompetes strain 100-23 by about 1,000-fold. So this is a competition ratio you're looking at. And if you inactivate the PKS cluster, now these strains are recovered at a 1-to-1 ratio, okay? So the presence of the PKS cluster now allows R12C to outcompete this 100-23 strain by 1,000-fold, which is dependent on the PKS cluster. And you can actually see a very similar trend in the different tissues. This killing, what we see, is not specific to strain R12C. We tested a large panel of Rotarrized strains. And again, here we look at the competition ratio. And this black bar here on the top indicates the competition between R12C and the test strain that you see on the x-axis. And this black bar here indicates that R12C outcompetes this test strain by 10 million-fold. However, if you inactivate the PKS cluster, now we're looking at the red bars, we actually now see that in this example of CR, we see that the strain is being recovered at a 1-to-1 level. So the competitive advantage is gone. And without going through all of these individual, you can already see that all these black bars are sitting, most of these at least, are sitting much higher up in the graph than the red bars. And that simply means that PKS here is responsible for outcompeting the competitive strain, right? Because that's why you have these high levels. Not for all strains, though. We found that in another probiotic strain, so this is strain 6475, again, applied in humans, has been shown in a clinical trial to promote bone health. And this strain has a range of probiotic activated molecules that have been characterized, at least in preclinical models. Rotarin is one example, and also reduction of colon cancer by the production of histamine. But this particular strain here actually doesn't care if PKS is around. We recover similar levels. The similar ratio is recovered if you co-incubate this R12C strain with 6475 in the absence or presence of PKS. So it does not matter. The strain 6475 strain is not impacted. And we wanted to know why. Why do some strains actually, can they coexist with strain R12C? So we've done comparative genomics, and these studies revealed that we found actually a gene cluster that was unique to strains that were completely resistant against PKS-mediated killing. And the one thing that you need to know for now is that one gene in particular caught our attention, which is an acetyltransferase. And in select microbes, actually, an acetyltransferase can provide resistance against lysozyme, but also select antimicrobials that target the peptidoglycan. So this became our target gene to investigate, to see if this gene, unique to strains resistant to PKS-mediated killing, if that played a role in that resistance. And to address that question, we come back to our genetic tools again. We inactivated the ACT gene, and we repeated the competition. And this is here in vivo data. I'll skip here the in vitro data, but here the in vivo data shows that PKS mediated killing does not happen in the presence of ACT. But if you inactivate the ACT gene, you now see that R2LC with PKS has the advantage. So this shows you here that the ACT provides protection. So for example, in the stomach, if we just look at that, we looked at different sections, we see here the competition ratio between the R2LC PKS producing strain and a strain that is resistant. I mean, there is some difference, but we're not looking at a millionfold here, right? And if you now inactivate the ACT gene, we see that our resistance actually is dropped, and now the strain becomes sensitive, showing in vivo that this ACT gene contributes to protection against PKS-mediated killing. And these differences that you look at here nearly are abolished if you inactivate PKS. So also telling us again that these differences here are PKS mediated. We then went on and had a closer look at the peptidoglycan, or the cell wall components here, and looked specifically at the acetylation. And Dr. Tokmo, who was a postdoc in the lab, led this work, and with the help of Dr. O, a research scientist in my lab, for about 10 years now. And what they had found is that this ACT gene promotes acetylation, so is responsible for acetylation of these cell wall components. And what we think is the case, and I will not go through all these numbers here, but you can take my word on it, that we demonstrated that overexpression of ACT increases the acetylation, and inactivation of ACT in a strain that contains ACT reduces the acetylation. And we think that this peptidoglycan acetylation contributes to the protection. We don't know how that part specifically at the molecular level works, but that is actually for a future study to figure that out. So really, in summary, and I think this really cute cartoon here summarizes it, whereby you have a pigmented strain with the PKS belt that is fighting an organism that encodes the ACT gene, but this organism here in the purple is able to fight off the PKS producing strain, is able to give it a match, so to speak. In contrast to this sad organism here sitting in the corner of the boxing ring, who does not code for ACT and is greatly outcompeted by this PKS producing strain. So these are rather specific, but I think it's a nice mechanistic study that provides us with a bit of insight with regards to how a particular organism can thrive in the gut, and what the underlying mechanisms are that contribute to outcompete other organisms, and how select probiotics can protect themselves against these actions of a secondary metabolite. And if there's really one thing, that would be great, if that would be the takeaway from this, is actually the message that strains matter. It really matters with what strain you are working with. I'll come to the end here. I am very grateful for the support of federal and non-federal funding agencies, and really I would not have been here without a fantastic team of scientists that were so brave to join my lab and to work with me, and they're really all doing a great job, and I'm grateful for that. And with that, I'll thank you very much for your attention, and thank you very much for listening. Thank you.
Okay, thank you very much, Dr. Van Pickeren, for your insight in your laboratory research about Lactobacillus reuteri and its potential benefit. Moving on, we are going to listen to Mr. Hirata. Before we are listening to his presentation, I will give you brief information about him. Mr. Haruhisa Hirata, PhD, currently is the Director Audit Supervisory Committee member of Wakamoto Pharmaceutical Japan. He graduated from the Department of Agricultural Chemistry, Kyoto University, and received a PhD in engineering from Osaka University in the field of fermentation technology. Mr. Hirata developed several probiotic products, namely Lebanon capsule, Lolon antidiarrheal, Meiji Probio yogurt, Wakame Teddy tablet, Advent-based toothpaste, and also oral tablet. Mr. Hirata was a former counselor, director, auditor, at the Intestinal Microbiology Society Foundation and also served as an auditor in Japanese Probiotic Association from 2013 to 2017. Without further ado, I invite Mr. Hirata to start the presentation. Mr. Hirata, you have 30 minutes. Please, the floor is yours.
ありがとうございます。丁寧なご紹介をいただきましてありがとうございます。若本製薬の平田です。ちょっと画面共有します。本日はこのご講演の機会を与えていただきありがとうございました。チェアパーソンのベニール・キート博士やその他の関係者の皆様にお礼申し上げます。このセミナーでは日本の100年を超えるプロバイオティックプロダクツの歴史、特に医薬品の開発の歴史を振り返ります。続いて、私たちが研究開発しました2つの新しいプロバイオティックプロダクツ、胃の健康に役立つヘリコバクターピロリ菌を抑制するプロバイオティクス、口の健康に役立つ歯周病菌や虫歯菌を抑制するプロバイオティクスです。で、最後に日本のプロバイオティクスの研究開発の現状と、将来に向けた課題と展望について少し述べたいと思います。まず最初に私が所属します若本製薬について簡単に紹介します。1929年に設立されまして、違う。最初は乾燥ビール酵母をタブレットにしたものです。で、錠剤の形で提供してました。1936年に、今日本の国菌とされてますアスペルギルソリゼを穀類培地に個体培養したものを成分として追加しまして、消化の効能ですね、消化の効能を追加しました若本錠を出しました。続いて1953年に、乳酸菌のストレプトコッカスペクリスを穀類培地に個体培養したものを追加しまして、成長の効能を追加しました。1962年に、今も販売しております協力若本の形になりました。この時点ではビタミン B 群の B1、B2、B6 を強化いたしました。その後、若本製薬としましては、ここに挙げましたようないくつかのプロバイオティック医薬品、あるいはプロバイオティック食品を研究開発しまして、今日はですね、この赤い字で示しました、東海大と明治乳業と共同開発しましたヨーグルト、2000年に上市したものについて、この下のサプリメントですね、ラクトバチルス、サリバリウス、WB21 を含むサプリメントの研究開発について紹介いたします。日本のプロバイオティック医薬品は、出ないのか。医療用医薬品と薬局用の2つに分かれます。医療用は通常の細菌を成分とする医薬品と抗生物質耐性の細菌を成分とする医薬品があります。薬局用は薬局で販売する OTC 医薬品と、規制緩和によりコンビニ等で販売できるようになった医薬部外品があります。 日本で最初のプロバイオティクス医薬品でありますビオヘルミンは1917年に製造販売されました。これは1907年にメチニコフがブルガリアの長寿の原因がヨーグルトの乳酸菌による腸内の腐敗菌の抑制と考えて論文を発表した10年後のことです。ビオヘルミンはこの発表に影響を受けた神戸の医師らが乳酸菌製剤の研究開発を行って生まれた医薬品で、ストレプトコッカスフェカリスとバチルス・サチルスを配合したものです。現在もこの形で販売されております。その後、ビヒドバクテリウムやクロストリジウム・ウチルカムを成分とするプロバイオティック医薬品が開発されました。抗生物質の服用が盛んになってきました。1970年頃に抗生物質による下痢や便秘の副作用の軽減を目的としまして、日本では抗生物質に耐性を付与した、ここでは AR と言いますが、失礼しました。AR を含むプロバイオティック医薬品が開発されました。1969年にエンテロノン R、1970年に若本製薬のレベニンが開発されています。で、これら多数のプロバイオティック医薬品が日本では当時ありましたが、医薬品の GMP と規制が進むの中で、これらの医薬品の再評価と、それからいろんな規格や効能がありましたが、それを統一するということが進みまして、1984年に、効能が統一されました。効能としましては、通常の細菌のものは、腸内フローラの異常に伴って生じるいろいろな症状の改善になります。抗生物質耐性のプロバイオティクス医薬品の効能は、抗生物質や化学療法剤を使用中の腸内フローラの異常に伴って生じる諸症状の改善。どちらもお腹の症状ですね。便秘、下痢に対する効能を明確にいたしました。それと並行して、これらのプロバイオティクス医薬品の原薬の規格や試験方法が1989年から1991年に日本薬局法外医薬品に収載されました。これが日本薬局法外医薬品に収載されている日本のプロバイオティクスの6つの成分です。上の5つは通常の細菌、一番下は抗生物質耐性の乳酸菌になります。一番上のラクトミンはストレプトコッカスフェカリス、ストレプトコッカスフェシウム、ラクトバチルスアシドフィルス、ラクトバチルスブルガリカスを含んでいます。ビヒドバクテリウムはビヒドバクテリウムロンガム、ビフィダム。ラクトバシルスカゼはラクトバシルスカゼ。有胞子乳酸菌ではバチルスコアギュランス。糖化菌アミロリティックバチルスではバチルス、サチルス他の菌種。で最後の抗生物質耐性乳酸菌につきましては、ここに示しましたような複数の乳酸菌が認められています。この薬局法外医薬品規格の内容ですが、6つの先ほどの6つの成分とも同じ様式で記載されております。まず製造方法ですね。 培養して集菌して乾燥して、乾燥でんぷん等と混ぜて原薬とします。2番目の定量につきましては、生菌数を測定します。ラクトミンの場合、規格としては 1×10 の8乗から 2×10 の12乗グラムあたり含むものが規格になっています。で3番目、正常ですが、白色からわずかに黄褐色のパウダー。同定につきましては、グラム染色や乳酸酸性を調べて同定しています。純度は重金属とヒ素の上限規格が決定されました。その他に、乾燥原料、貯法、あるいは圧生のための培地が規定されています。で、これらはプロバイオティクスバクテリアの基準ですが、当時、1989年頃にフラーが定義したように、プロバイオティクスは赤字で示しましたように、腸管の微生物のバランスを改善するという言葉が入っておりましたように、日本でもプロバイオティクス医薬品の、大部分のバクテリアは、ここの下に田中がまとめました8つの基準に合っております。1つ目が常在菌であること、2つ目が上部消化管のバリアに抵抗性であること、3番目が下部消化管で、小腸や大腸で増殖できること、宿主に有益な影響を与えること、3年以上生きて保存できること、副作用がなく安全性が高いということ、で服用しやすいものであること、さらに原価が安くて価格も安いという、このようなやつの基準を満たしたプロバイオティック医薬品が日本では当時から製造販売されております。さて、OTC と医薬部外品を一例をここに示します。下の段に示しましたのが、2004年に OTC 医薬品の中からさらに作用が緩和で副作用がない医薬品を医薬部外品として指定されました。協力若元もこの真ん中にありますけれども、医薬部外品に、日本では医薬部外品になって、コンビニ等の薬局以外でも販売されております。上の段が、いわゆる成長薬と言われている OTC ドラッグです。いくつもありますし、2007年に OTC 医薬品が3つに分類されたときに、これらの成長薬はいずれも安全性が高いものとして、第3番目の分類になっております。ここからはプロバイオティックフーズの歴史について少しお話しします。私は個人的には納豆をですね、大豆をバチルス、サティルスで発酵させたのが日本のプロバイオティクスの初めのものではないかと考えておりますけれども、これは文献には1050年ぐらいから納豆という名前が記載されております。乳製品を使ったプロバイオティクス食品としましては、1919年にカルピスが、これは殺菌したものですが、発売され、1935年にヤクルトが発売されました。その後、1971年に明治が 最初のプレーンヨーグルト、ラクトバチルス・ブルガリカスとストレプトコッカス・サーモフィルスで発行したものです。発売しました。この2年後の1973年に明治はブルガリア政府の許可を受けまして、この名前をですね、明治ブルガリアヨーグルトに変えて、これも今日本のヨーグルトの代名詞としてたくさん販売されております。で2000年にはプロバイオティックヨーグルトの先駆けとなります明治プロビヨーグルト LG21 が販売されています。でこの間にですね、日本の食品の表示の基準が2つ、プロバイオティクスに関係する基準が2つできました。1991年に特定保険用食品、当時厚生省、今は消費者庁で審査していますが、2015年に機能性表示食品、これは消費者庁が作った、これらの食品の表示が新しく認められるようになりました。ここからは2つの新しいプロバイオティクスの私たちの研究開発を紹介いたします。これが明治プロビロヨーグルトですね。2000年の3月から2020年の9月まで、これ明治のホームページでは82億個以上販売されたということです。ヘリコバクターピロリ感染の話を少しいたします。ヘリコバクターピロリは、胃炎、胃潰瘍、十二指腸潰瘍、胃がんの原因菌です。1979年にオーストラリアのワーレンとマーシャルが、それまで共産性のために微生物はいないと思われていました人の胃の中に発見した細菌です。その後、ピロリを除菌する医薬品の研究が行われました。彼らはこの業績により2005年にノーベル賞を受賞しました。日本は胃潰瘍、胃がんが非常に多い国ですが、ピロリ菌の中でも特に病原性の高い東アジア株というものに感染者が多いことが原因です。このグラフにありますように、1974年には国民の半数以上がピロリ菌に感染していました。2000年からアモキシシリン、クラリストロマイシン、プロトンポンプインヒビターの3剤併用によるピロリの除菌治療が保険適用になりまして、そのこともありまして、2008年、一番右端の2008年には感染者は激減しておりますが、まだ感染者ゼロになるのはほど遠い状況であります。これが我々がヘリコバクター感染に対して有効であるとスクリーニングしました乳酸菌です。左のグラフはラクトバチルス・サリバリウスという菌株が最もマウスの胃の中のピロリ菌を抑制する作用が強いことが分かりました。左側のバーが、胃内のピロリ菌数、右側が血中のピロリに対する抗体価ですが、ラクトバチルス・サリバリウスの投与によってどちらも低下しております。右側のグラフが LG21 ヨーグルト、最初ラクトバチルス・サリバリウスという菌株を見つけたんですが、その後このラクトバチルス・サリバリウスをどんな形で人々に届ければいいかというのを考えまして、ヨーグルトの形態が適しているというふうに考えて、国内のヨーグルトメーカーに共同開発を呼びかけて、明治乳業、当時の明治乳業、今の明治と一緒に研究を始めました。その中で、ラクトバチルス・サリバリウスはヨーグルトの中での安定性があまり良くないので、明治で選抜したのが LG21 という菌株です。これが LG21 をヨーグルトを最初に投与した臨床研究の結果です。右のヨーグルトプラス LG21 群では、胃内のピロリ菌数の指標であります尿素呼吸試験の値が優位に低下いたしました。LG21 に関する臨床研究は、これまでに12法の論文になっております。一部を紹介しますと、一番上が、12週間の摂取で、ディスペプティックな症状が軽減されたというものです。真ん中は4週間の LG21 の投与によって、散剤併用療法では耐性菌が出現しておりまして、除菌率が低下しておりましたが、LG21 ヨーグルトを除菌療法の4週間前から摂取していただくことで、除菌率が 82.6%、プラセボは 69.3% で優位に上がりました。一番下が、これはちょっと変わったタイの幼稚園児を対象にした試験です。LG21 の入ったチーズを12ヶ月間食べていただきまして、12ヶ月後に、82例中24例が除菌されたと。それから感染していなかった幼稚園児の方の自然感染ですが、プラセボでは 8.1% を1年間で感染しましたが、LG21 群では 4.1% に抑えられたという結果です。次にアバンビーズオーラルタブレットの紹介をいたします。2017年4月にラクトバチルス・サリバリウス WB21 とキシリトールと緑茶カテキンを配合したアバンビーズオーラルタブレットを発売しました。現在、e コマースの販売ルートで販売しております。これはですね、2004年にワカメイト D タブレットという名前で、ラクトバチルス・サリバリウス WB21 にキシリトールを加えたタブレットを配合したものを発売したんですが、薬局ではなかなか売れませんでして、若本製薬では発売を中止しました。しかし、その効果に着目された福岡市の歯科の先生が非常に興味を持たれまして、現在ではその歯科の先生の歯科医院の専売品として、ワカメイト D も現在も販売していただいております。これが日本の歯周病の罹患状況です。下が年齢階級で、横軸が罹患率になりますが、赤で示しました歯肉の出血ですね。歯肉の出血につきましては、もう左端の15歳から24歳ぐらいから 40% 前後の人が歯肉の出血が認められます。青で示しました歯周ポケット 4mm 以上の人は、これは年齢とともに割合が高まりまして、65歳から74歳ですと、6割の方が、こういう症状を示しております。歯周病が悪化しますと、歯を失ってクオリティオブライフを低下するだけでなく、心疾患、脳血管疾患、糖尿病も悪化することが明らかとなっております。歯周病は歯周病菌、虫歯は虫歯菌によって起こる口腔内感染症ですので、歯周病菌、虫歯菌を抑制して口腔環境を健康にするプロバイオティクス食品の研究開発を行いました。これが、歯周病菌であるポルフィロモナス・ジンジバリスに対する増殖抑制効果を示しております。ラクトバチルス・サリバリス WB21 は、単独で ATCC、JCM、この2種類の歯周病菌の増殖を抑制しますし、さらに緑茶カテキンを一緒に併用しますと、その効果が高まることが分かりました。データはここでは示しませんが、 WB21 は虫歯菌の増殖、不溶性グルカン酸性に対する抑制効果も認めております。WP21 の臨床研究も6法あります。簡単に紹介しますと、一番上と真ん中は東北大学で実施しました歯周病の指標に対する改善を調べたものです。約7億個の WB21 を含むタブレットを8週間摂取していただいた結果ですね、特に、喫煙者は歯周の状態が良くないということが分かっておりますが、喫煙者のグループにおいて、プラークインデックスやプロービングポケットディプスですね、歯周疾患の指標が改善されたという結果です。2番目が、この改善がどういう基準によるものかというのを調べるために、口腔内の細菌層について調べたものですが、摂取によりまして、歯周病原菌の、歯周病原菌はたくさんありますけど、この中では5種類の歯周病原菌の総数を WB21 は減らすことが確認されました。一番下は口臭に対する効果を調べたもので、これにつきましても口臭が減少するという結果を得ております。ここから日本の食品の表示の話をいたします。消費者庁のホームページにあるもので、非常に見にくいものですが、一番下、フーズインジェネラルというのは一般の食品で、表示、プロバイオティクスの表示に関係している食品としましては、真ん中のフーズ・フォー・スペシファイド・ヘルス・ユースですね。特定保険用食品。で、その上のフーズ・ウィズ・ファンクション・クレームズ。機能性表示食品。この2つが特に機能性あるいは特定保険を表示できる食品として日本では認められています。まず、特定保険用食品につきましては、現在消費者庁で審査されて承認されると、こういう表示ができる食品になりますが、これは、その食品に関する臨床試験の成績、安全性、食経験、規格等の必要な資料を消費者庁に提出して、承認されれば表示が認められる食品になります。2022年現在、1060品目あります。市場は3000億円と見込まれています。保険の用途としては、インドネシアの先ほどの話と同様に、お腹の調子を整えるヘルスユースが多く販売されております。プロバイオティクスとしては、乳酸菌、ラクトバチルス、ビヒドバクテリウム、ストレプトコッカスなどの14菌株が、特定保険用食品には認められています。続きまして、2015年に消費者庁が作りました機能性表示食品です。この研究開発は非常に盛んに行われておりまして、これは、機能性を裏付ける臨床試験、疫学データ、安全性、食経験、規格等の必要な資料を消費者庁に届け出て、消費者庁が受理すれば、販売者の責任において、これらの表示ができる食品です。現在5485品目、4400億円の市場が見込まれております。プロバイオティクスとしては、乳酸菌が229品目、ビフィズス菌が153品目。例えば、下に挙げたような新たな機能性表示がされております。
Hirata,
はい。
I'm
Yes.
Mr. Hitarta, I
Yes.
apologize for the interruption, but please can you make it a little bit faster because the time is almost over. You have 5 minutes more for your
はい。
presentation. Thank you.
わかりました。はい。はい、早めに行います。すいません。ここに挙げたような新しい機能が認められています。認められた食品が販売されています。最後に、今のプロバイオティクスの日本の現状ですが、機能性表示食品はたくさん開発されておりますが、医薬品の開発は遅れております。日本の製薬会社が主体となって、2017年にジャパンマイクロビオームコンソーシアムを設立しまして、現在31の会社、食品会社も含めて31の会社が共同で、プロバイオティクス開発に必要な研究開発を行っております。で、今年の2月25日にようやくですね、日本でも、これ2016年にアメリカの FDA がインダストリーに向けたスペシフィックガイダンスという形で出されているライブバイオセラピューティックプロダクツのレポートが出されました。日本の医療薬品を審査する PMDA の科学委員会において作られたものです。LBP の研究開発のステップとしましては、ここに挙げました5つの菌株の選出、選抜、品質の特性を調べる、製造方法を開発する、で、非臨床試験、臨床試験を行うということです。まだこれは、官と学の間のもので、産業界に対するスペシフィックガイダンスのような形にはなっておりませんので、これをより良くしていくことが課題だと考えております。最後に少し時間がかかって恐縮ですが、日本の100年を超えるプロバイオティクスプロダクトの開発の歴史について述べました。それから、明治プロビオヨーグルトとアバンビーズという新しい機能を持ったプロバイオティクスプロダクトを紹介いたしました。最後に、日本のプロバイオティクスの開発の現状と、今後の課題と展望を述べました。以上です。どうもご清聴ありがとうございました。
Thank you very much, Mr. Hirata. Now we are moving to the discussion session. The discussion session mainly will be the question and answer between panelists and the speakers because of our time limitation. For the first panelist, I will invite Ibu Rahmi Steorini to give a feedback and also maybe some question to the speakers. Please, Ibu Ririn, time is yours.
Pertama siapa namanya yang dari
Ya, thank you for Ibu Wiwin. Terima kasih. Ya, terima kasih untuk for your presentation, Mr. Van Peekran. In your presentation, FDA has not approved a probiotic for a specific head climb. What is a specific head climb? Can you give an example? Because in USA pharmacies, I got probiotic with claim for emotional
ada yang offline juga ini.
And how to guarantee the number of bacteria is correct. Okay, thank you.
Thank you, Ibu Rahmi. Maybe we will have a feedback first from Dr. Van Peekran, please. Dr. Van Pitkern, can you hear us here from Indonesia? Hello, Dr. Van Pitkern, can you hear us? I think we have some technical difficulties. Maybe Bu Rahmi can give another question for Mr. Hirata first. Please, Bu Rahmi.
Baik, terima kasih kepada Bapak Hirata. Ini pakai bahasa Indonesia saja ya, Jay. Apa perbedaan probiotik yang masuk ke dalam OTC drugs dan yang masuk ke dalam QC drug? Terima kasih.
Thank you, Bu Rahmi. So, Mr. Hirata, the question from Bu Rahmi is, what is the difference?
はい。もともと OTC 薬品の中で、2004年に非常に長い歴史があって安全性が高い OTC 薬品の中からいくつかの、協力若元も含めていくつかのプロバイオティック医薬品 OTC 医薬品が、医薬部外品になりました。それは薬局以外でもお客さんが求められるようにということで規制緩和の中で進んだもので、上の OTC 医薬品との違いといいますと、やはり長い歴史のあるプロバイオティック医薬品が選ばれたということになると思います。
Thank you, Mr. Hirata. We will try to connect again with Dr. Van Pitkern. Good evening in Wisconsin. Dr. Van Pitkern, can you hear us? Okay, I can't hear your voice. I think you can hear my voice, but we are in Indonesia cannot hear your voice. Can you try again, please?
Okay, can you hear me now?
Yes, it's very clear. Thank you very much.
Thank you. Yeah.
So our first question from you is from Ibu Rahmi. Ibu Rahmi, maybe you can repeat your question, please.
Thank you. Baik, saya akan mengulang pertanyaan saya. In your presentation, Mr. John, you explained that FDA has not approved a probiotic for a specific head claim. What is a specific head claim? Can you give an example? Because in USA pharmacies, we got probiotic with claims for emotional well-being relaxation, digestive balance, and contain 16 probiotics. How to guarantee this claim is true and how to guarantee the number of bacteria is correct? Thank you.
Thank you very much for your question. And there were a couple of questions embedded in that. So first of all, the question, what is a health claim? A health claim is considered a statement that refers to curing a disease or ameliorating a disease. So for example, an example of a health claim would be to reduce intestinal inflammation, because then you are referring to the inflammation as the disease part, and that would be considered a health claim. The claims that you were describing are considered so-called structure function claims, is my understanding. And what that means is that none of these claims, these so-called structure function claims that currently are described in the US, none of these structure function claims are linked to a particular disease, but are actually more a general well-being. So an example of that would be promotes bone health, or an example would be of promotes digestive health, this probiotic. So it's very general, and the real evidence for that, the hardcore evidence, I'm not sure for these structure function claims what that would be, other than, of course, a clinical trial that would underlie that. But I am not sure if that needs to be put on the table, the data for a clinical trial, in order to support a structure function claim. Then you also had a question how to guarantee the actual numbers. That typically is done in assays, whereby basically stability assays, whereby or shelf life study assays, whereby it is the products are incubated at a particular condition, and your samples are taken over time, whereby the viability of the product, so basically by typical standard plate counts, is determined, and that will determine the viability. And Dr. Hirata already alluded to a 3-year shelf stability, and it will also be under similar assays that would be determined to determine that viability.
Thank you very much, Dr. Van Peetskeren. I think it's very clear about your answer. Okay, the next panelist, I will invite Dr. Rina Agustina to give some feedback, and also if you have any question for our speaker.
Yeah.
Dr. Rina, please, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Madam Moderator. Can you hear me well?
Yes, we can hear you clearly.
Yeah, thank you for an excellent presentation for both speakers. First, I would like to ask maybe more questions to Professor Van Peetskeren. Sorry to spell it wrong, maybe.
That's okay.
Okay. Again, I will continue the question on the structure function. I understand you mentioned that it need to reconfirm, but what would be the requirement? The first one, when we would like to declare support digestive health, and you explain about the competitiveness and the metabolic role production, would that a good indicator or biomarkers for support digestive health? Would that be competitiveness or metabolic role, recovery of the strain, or antibiotic direct antagonism or immunomodulator? What would be the most biomarker or indicators that support that structure function? And the second question is that you actually presented one of the study of Dwar et al. 2017. And it shows that there's a different the play of these gainers, some gainers toward the specific age, location of the gut. The question will be how we can extrapolate these strain findings to different subjects, and what would actually that allow to be extrapolated, and how actually the minimum requirement for extrapolation, or is there any nonsense to really extrapolate those findings? And the third questions on the PKS productions is really important study. Congratulations for that. And the question will be on because how important is the role on the aryl hydrocarbon receptor AHR, which is actually very important for some metabolic or inflammatory or mucosal immune system. So what would be the basal level of the AHR activation, especially when we want to consider the PKS role in each of the strain? So what would be the strain needed to really have the the basal level of the AHR activation? Would that be actually only be contributed by the PKS role? And the next question will be, and how this will link to the potential amelioration of many inflammatory markers or inflammatory problem like the colitis or diarrhea, which we know that some of the findings from the uteri very much linked to these particular problems. And the last question will be on the fecal recovery, because you mentioned about the fecal recovery, and then show that there's a different strain potential on fecal recovery. For example, on the same type of probiotics given, or perhaps the CF would the same, but they comes up with different recovery. So the question whether it is really determine the survival between the strain, but somehow it is really different, the result of the in vitro study and the result of the observed human study. There's some differences, and whether those who are not being recovered is really not providing any survival capacity, while some of them do not recover much on the fecal sample, but they do give a substantial effect on the human outcomes. So that's actually my questions to the first speaker. Shall I continue the second one, or will be feedback from the professor first?
Please, Dr. Victor.
Yeah, that's a lot of questions to process here, and I'm very happy to address them. But please remind me if I forgot one or not. So, starting off with the structure function and how actually the survival of a probiotic in the gut can be linked to structure function. I think that really is linked to, and the importance I think of the work that we have done is really linked to understanding how select strains can survive in the gastrointestinal environment, because ultimately the organism needs to be able to survive GI transit if we want to put it under the umbrella of a probiotic, at least if we take into consideration the definition that is currently out there. So this work will not only help us understand how select strains can compete and have this antagonistic effect against other strains, that is one, and we now know how, which is through PKS. It also provided on the other end of the spectrum insight into how other select strains, like the strain that was resistant against this killing, how that actually can still coexist and persist within this within the same ecosystem. So really it provided two pieces of knowledge, so to speak, one with regards to the antagonism and the other one how it actually can compete with the organism. And ultimately I think that this information will be important, especially when you think about probiotic mixtures and cultures, because you want to actually have strains that are compatible, and you don't want to have a mixture whereby one strain is automatically killing off another strain in your product, right? So I think that actually will be helpful in the design and the development and the selection specifically of probiotics. In general, I mean, I personally am a fan of mechanism. I think it helps for the aforementioned reasons, but ultimately, I mean, mechanism is not always important to understand mechanism while you still can deal with a probiotic, but it really will help the selection process and to leverage that knowledge for engineering purposes to develop what I call next-gen probiotics. Another question you had, and I may not do them in order, but you can remind me if the ones that I forgot. Another question you had was with regards to this PKS cluster and the activation of the aryl hydrocarbon receptor, and this is correct. So I've not talked about it in this presentation, but indeed we had another study showing that this exact same PKS cluster activates the aryl hydrocarbon receptor. And a follow-up question that you had was how this actually is linked to immunity and the levels of activation that would be required to have the effect. In terms of how this actually can impact immunity and colitis, it's not published, but we actually have unpublished data showing that the activation of the aryl hydrocarbon receptor helps to prevent the development of colitis in a mouse model. So that is at least preliminary data there in a preclinical model. The specific level of activation that is needed, that really is unknown. And it's an excellent question, right? And because how much is needed? How much is really needed to have the effect? And I think ultimately that will depend on the individual. I think it will depend on the immune status of the individual. It may depend on actually the composition of microbes that are there. And it will, and actually, and I think that work that I presented will demonstrate that, because it also depends which other antagonist may be present in the gut microbiome that can actually impact the probiotic itself again and can impact its survival. And therefore, you know, depending on who's there and what the immune status is, it may actually depend on how much is needed. So that is a very a tough question to answer. Actually, I cannot answer it because of these reasons here. And that needs to be validated. In terms of the level and how the immune system is activated through AHR and how it can fight off inflammation, well, one aspect of AHR, what has been demonstrated, is that activation of the irohydrocarbon receptor, which is a ligand-activated transcription factor, that plays a role in mucosal immunity. And specifically what happens is that dietary ligands, for example, can activate AHR. Think of the brassica vegetables that are known to activate the irohydrocarbon receptor. Tryptophan metabolism by L. Roiteroy, really elegant study by Zelante is the first author, published a couple of years ago, showing that the metabolism of tryptophan by Lactobacillus reuteri actually activates the irohydrocarbon receptor and thereby fights off a mouse pathogen in the gut. And our work showing that this polyketide synthase cluster can activate the irohydrocarbon receptor. And what all of these activation pathways have in common is that this activation leads to the activation of the production of interleukin-22, which is a cytokine. And the activation of interleukin-22 has as a downstream effect that it subsequently activates Rec3 beta and Rec3 gamma, which are small antimicrobial molecules produced by the mammalian host. And these play a key role to fight off bacteria that otherwise may actually go into sepsis or into the bloodstream. And the IL-22 it depends on the disease model, but it can actually suppress intestinal inflammation overall. So that is my response to that question. Another question you had was with regards to how we can explain these differences in the composition by the study of Duar and the other study of the microbial composition, the cocktail that goes in and who survives and who's not. Again, that all depends on the interpersonal variability, and that will all depend on the composition of the gut microbiome again. And linking it back again to the presence of antagonists being present and whatnot. So I think that we have to be, and this is my opinion, I think that we have to be really careful in putting forward a probiotic and that fixes everything and it's fine, and because we will have the situation there, it will be dependent on host factors, and it will be dependent on the microbiome. So I think moving forward and thinking of the future of probiotics, it is my opinion that we really cannot view this probiotic as an individual organism anymore, but that we really need to view it, and we need to take a holistic approach, thinking about the probiotic within the mammalian system and how it interacts with other microbes, and what we can learn from these studies to subsequently put forward and have better selection procedures for probiotics. Without doing that, I think that if we don't take this holistic approach, and if we keep viewing probiotics still at the level as we do it now, as often as individual strains, we will end up with more and more clinical trials that fail, I think. And taking the holistic approach and understanding mechanism will ultimately help and steps forward towards a better selection and greater chance of success for probiotics, I think. Did I miss any of your questions that you asked?
Yeah, the last one about the fecal recovery.
What was your question on the, can you please repeat the question for me on that?
Okay, on the fecal recovery, we know that even they give the same amount of the CFU amount of the probiotics, they different types of strain, they may recover differently, right, in the fecal sample.
Yeah.
So, there's some also studies showing that even if they are not recovering in fecal sample, but they actually giving a substantial effect on some of the human outcome, it is actually already kind of a good sign. So it doesn't really, it means that fecal recovery doesn't really mean linear to the survival. So what do you think? Because it's really used a lot, the fecal recovery as a marker to show the survival of the probiotics, which actually not very much consistent between one study to another.
Yeah, I think that you will enter a bit of a gray area if we think about probiotics and what they are and what the current definition at least is. And what you explained to me, I'm nearly thinking of the application of postbiotics nearly to some degree, whereby it has been nicely shown in some studies that dead microorganisms or inactivated microorganisms can still have a health benefit. So I think technically you can have, if that's what you're alluding to, at least, you can have a probiotic that is viable on the shelf and you take it in and it dies through GI transit, and thereby you're not recovering it in the fecal material anymore. Yet you technically, yes, you can have a health promoting effect. I don't know enough about the definition, if that still would actually be considered a probiotic, because you start off with a live microorganism. But I at least my indication of this is, but I suggest you have this clarified by Professor Salminen from the ISAB, that it needs to survive GI transit to have that effect there. But yeah, it certainly can have a health promoting effect, but I don't think it then would be considered that much of a probiotic anymore. The other thing that now I have to throw for one more minute maybe, that I want to emphasize, that I forgot to mention in my presentation, and I alluded to strain specificity, is the other main point is that probiotics often actually do not colonize. It is a common misconception that the intake of a probiotic will automatically lead to colonization. And I think that's a really important point to make, because the is that often probiotics do not colonize unless the ecological niche is available. So someone who has not undergone antibiotic treatment, and if it would be considered a diverse microbiome, for example, and if I were to take, I'm not on antibiotics now, and if I were to take a probiotic, it's likely that probiotic will actually colonize in my gut. Again, unless that niche is available. And really elegant work by Jens Walter and Douwe van Sinderen has been done, published a couple of years ago in Cell Host Microbe, showing that individuals taking in a probiotic bifidobacterium, that often that bifidobacterium does not colonize, and they identified that the reason why that bifidobacterium was not colonizing, because these individuals already had a bifidobacterium in their gut. In individuals where the bifidobacterium did colonize upon ingestion, originally there was no bifidobacterium present. So the idea is that really these microbes have select niches. If the niche is available, that can lead to colonization. And I think that, going about this Q&A and some important questions that you asked, and excellent questions, I just wanted to make use of the opportunity to highlight that they don't colonize and that there is strain specificity.
Thank you, Professor. Yeah, so there's still a lot of questions, but because of the time, may I continue with the question to Professor Haruhisa, Madam Moderator?
Dr. Rina, please make it a brief question, and maybe just one question for Dr. Hirata. Thank you.
Yeah, so thank you, Dr. Hirata. So It's very excellent information on the role of several probiotics on the suppression of this, to the Helicobacter pylori infection. So you presented 3 different studies which shows different duration of intervention. The other one is 12 weeks, the other one is 4 weeks, and the other one with 12 months with different types of the subject. But they comes up with different different effect size. So what would be the link to the particular information on the regulatory in terms of the duration of the study? This actually really dependent of that duration or dependent on the particular strain or subject recruited. Thank you.
Yes, Mr. Hirata, your feedback.
はい、臨床試験の期間、接種の期間の話だと思いますけれども、特に決まりはありません。医師の先生方は、これぐらいの期間接種すると、プラセボ群と比べて効果が期待できるという形で研究を進めています。そのため、紹介しましたタイの研究では、1年間という非常に長い接種期間になりましたけれども、通常は、食品として摂取、ヨーグルトを食品として摂取する場合は、長くても12週間ぐらいの試験が多いと思います。以上です。
Okay, thank you very much, Dr. Hirata, and also Dr. Rina and Dr. Van Viskasen. Now we are moving to the third panelist, Dr. Rini. I invite Dr. Rini to give feedback and maybe some question, please.
Thank you. Thank you for giving us the time. Well, as I'm coming from the commercial sites, so maybe the question will be more to the commercial or industrial aspects. So my first questions for Dr. Hirata. Dalam presentasi Dr. Hirata dinyatakan bahwa dalam Japan Microbiome Consortium ini telah ada beberapa riset yang dimana riset ini telah dilaporkan dan telah dipresentasikan pada PMDA's Scientific Committee, yang dimana hal ini sebagai referensi bagi PMDA dalam menyusun specific guidance for industry. Pertanyaan kami kepada Dr. Hirata, bagaimana ekspektasi dokter terhadap specific guidance for industry berdasarkan feedback atau berdasarkan input masukan dari riset yang dilakukan oleh JMBC. Demikian pertanyaan untuk Dr. Hirata.
Thank you, Dr. Rini. Dr. Hirata, you may answer the question.
はい、ありがとうございます。日本では FDA に遅れて、もう6年ぐらい遅れて PMDA の科学委員会で先ほど報告がなされたという紹介をしましたけれども、これがようやく出てきましたので、プロバイオティックの医薬品メーカーとしましては、この報告書に書かれた内容に従って、PMDA と相談しながらですね、開発を進めていけると、そういう意味ではある程度道しるべになった報告書だと考えております。以上です。
Thank you. Is that enough, Dr. Rini? Okay. Do you have other question for Dr. Van Pickeren?
Yes, one question.
Yes, please, Dr. Rini.
To Professor Van Pickeren. Having the scientific trials ongoing at your laboratory at VPL, I believe you would like to have a commercial product in the future as a result of your trial, which is perhaps you hope that your the product, the trial will be more health beneficial for more people and patients. Could you share with us what do you think? What are challenges to register a commercial product of probiotic with specific health claims later in US FDA?
Yeah, that is
Your
an
opinion
excellent.
as a scientist. Thank you.
Yeah, that is an excellent question. With regards to what my lab is working on mostly, and I think the closest we are with regards to bringing a probiotic to the clinic, these are actually engineered probiotics. So these are not the native probiotics that we talk about here, but these are probiotics engineered to produce a recombinant molecule that will alleviate certain disease markers. And with that specifically, there are multiple. One is, of course, the safety, the stability of the product in terms of going from culturing to a small volume to fermenter volume culturing to ensure the proper stability of the engineered protein. And of course the translation in itself to go from animal models to clinical trials, which really is the $1 million question. I think that I always have jokingly say, I mean, really most probiotics really are for mice, right? Are for mouse, because all these trials have so promising in mice, but the big challenge is, of course, to translate that to humans, and coming along with some of these shortcomings, if you like, and the hurdles that we need to overcome for that. So we are not there yet, but we are making great progress, and I'm really looking forward to the stage that we can actually do. And we do have, and I think that is important towards the development, we do have an upcoming trial in with one of our industrial collaborators in non-human primates. And I think that is an important step towards addressing safety. And in addition to work with disease models or animal models other than mice, I think will also help in the transition process towards humans. So and here we can think of pigs or non-human primates as that will be helpful in the transition of a probiotic from the lab, or an engineered probiotic, I should say, towards the clinic. And I think it depends on the application, but I think pigs especially can be helpful, because an advantage, you can feed a diet that humans eat, and because pigs nearly eat everything, and that can help actually with understanding the interplay between diet and between the probiotic and probiotic survival, etc. So I hope I hereby have answered your question. Thank you.
Thank you, Professor Van Pickeren.
Okay, thank you very much, Dr. Rini, Dr. Van Pickeren, and Dr. Hirata. Ladies and gentlemen, because of the time limitation, I have to close this first session because of the limitation. I apologize for the participant because we cannot read any of your questions. Once again, I would like to say thank you for the speaker, Dr. Van Pickeren, Dr. Hirata, and also our panelists, Ibu Rahmi, Dr. Rina, and Dr. Rini. Thank you very much for very interesting and meaningful insight about probiotic research and development for the first session.
Thank you, Ibu Moderator. Thank you, all the speakers.
Thank you.
Thank you very much. This is the end of the first session. I will give the session to the MC. Please, Ibu Pulan. Thank you.
Okay, thank you very much for the speakers, panelists, and moderator. Distinguished ladies and gentlemen, next we are going to the next session. The topic of the seminar is Regulatory Overview, Research and Development of Probiotic Product in Australia and Indonesia, with the speakers are Patricia Conway, BAc, MAc, PhD, and Professor Ingrid Suryanti Surono, MSc, PhD. Yes. This session will be facilitated by the moderator, Ms. Wiwin Heryati. Over to you. Thank you.
Thank you very much, Ibu Pulan. Because of the time limitation, we directly come to the second session. In the second session, we will discuss about the research, development, and regulatory overview of probiotic in Indonesia and Australia. In the second session, we have two speakers, which is Professor Ingrid Surono, MSc, PhD from the Indonesian Scientific Society for Probiotic and Prebiotic. Good day, Ingrid. Thank you very much for joining us today. And the second sorry, we have sound difficulties. And the second speaker is Dr. Conway. Good day, Dr. Patricia. Have you joined us? Dr. Patricia?
Australia. I can hear you. Can you hear me?
Okay. Thank you, Dr. Patricia. We can hear you clearly. Excellent. Thank you very much for joining us today.
It's a pleasure.
And also we have three panelists, which is Ibu Dwiana. She is the Director of Registration Traditional Medicine, Health Supplement, and Cosmetic, Indonesian FDA. Selamat siang, Ibu. And then we have a second panelist, I'm sorry for the delay. This and the second panelist, Bapak Raymond from Dexa Medica. Good day, Bapak Raymond.
Ya, selamat siang. Good afternoon, everyone.
Thank you for taking the time to join us today, Pak Raymond. And then our third panelist is Dr. Agnes from PT Kalbe Pharma. Good day, Dr. Agnes. Oh, I'm sorry.
Hello. Hello, everyone. Good morning.
Thank you very much for all the speakers and the panelists who have joined us today. We are moving on to our first topic, which is the regulatory overview, research and development of probiotic product in Indonesia. We have our speaker, Professor Dr. Ingrid. Before we are listening to Dr. Ingrid, I will read the brief CV of Professor Ingrid. Professor Ingrid Suryati Surono is a professor in functional food, Bina Nusantara University and the President of Indonesian Scientific Society for Probiotic and Prebiotic. She graduated from Food Technology Department, Faculty of Agricultural Technology, Bogor Agricultural University in 1980. She earned her MSc in Dairy Microbiology and Food Processing from ASEAN Institute of Technology, Bangkok, Thailand in 1984. And she also earned a PhD from the United Graduate School, Gifu University, Japan in 1996. She dedicated herself to probiotic research since 1992, and her research interests are in child nutrition, functional foods, and food safety. She was with Functional Food Forum University of Turku, Finland, as visiting professor in 2004 to 2006. Her scientific contribution gives significant impact in her field, as shown by Scopus Index 17 and Google Scholar Age Index 24 and her scientific profile scores. Without further ado, I invite Professor Ingrid to give the presentation. You have 30 minutes, Professor Ingrid. Please, the floor is yours.
Thank you so much. Allow me to share my presentation material. Okay, let me start then. Good day, everyone. Assalamualaikum warahmatullahi wabarakatuh. Let me start with the probiotic research on evidence-based efficacy and regulatory in Indonesia. Actually, the aim of my presentation is just to show you as a model that those requirements by the regulatory in Badan POM, we can fulfill from the in vitro up to the clinical trial. Okay, let's start with the characteristic of probiotics. We know that the probiotics should be able to pass through the digestive system and then able to attach the intestinal epithelial cell as well as colonizing, able to maintain good viability, and then able to utilize nutrients and substrates in a normal diet, non-pathogenic and non-toxic, and capable of exerting beneficial effect on the host. And probiotics are strain specific and dose dependent. Well, we start with the nomenclature of the probiotic. Yeah, the nomenclature of probiotic is genus, species, and strain. It's according to the International Journal of Systemic and Evolutionary Microbiology. So it's some of the species and strains change, yeah, the name, especially species. For example, Lactobacillus plantarum, it's become Lactobacillus plantarum. Yeah, we can check directly at the International Journal of Systemic and Evolutionary Microbiology. And then we have to know also the source of probiotic culture, the origin, yeah, of the culture when it is being isolated and identified. And then name of foods added with powder, liquid, suspension of probiotic culture, as well as supplements, yeah, in the form of supplements. Then what happened this? Oh, okay, yeah. So products containing probiotic bacteria, yeah, available in two forms, yeah, as the supplements and foods. We refer to the definition. Yeah, by definition, probiotic is live microorganisms that, when administered inadequate amount, confer a health benefit on the host based on FAO, WHO 2002 and ISAP 2013. So we have to identify the microbes and then assess the level of viable cells in foods and supplements in accurate way. That means the methods of enumeration should be also the best method. A sufficient number of live probiotics delivered in a product, then we can detect. And an evidence-based clinical trial with effective dose, and both regulatory and quality control authorities should have well-defined means to assess viable cells of probiotic in foods, in supplements, or in any substances regarded as probiotic, to ensure the consumers get the required dose of viable cells of the probiotic strains with each intake. Let's see the definition. It's been mentioned that inadequate amount, right? So then it's still a gold standard method to characterize the function of probiotic by knowing the ability of the microbes to survive in the GI tract, right? To remain viable in the GI tract. And then we have to do safety assessment and then clinical evidence, as well as viable efficacious dose throughout the shelf life. So then the viable cell counts, the viable bacteria of probiotic bacteria should be still in the efficacious dose at the end of shelf life. So then this is the authenticity of probiotic. Authenticity is defined strain, right? And then the viable counts at the efficacious dose, supporting by clinical trial. Then by this, now I think everybody here in the world try to do global harmonization of regulation, together with industry, health professionals, researchers, consumers, and society. Of course, the authority of each nation try to do the harmonization. Probiotic health benefits are strain specific and dose dependent. When that means we need to consume correct strain at the recommended dose, essential to achieve probiotic health benefit. Right? Then the reliable methods needed to facilitate probiotic strain identification and enumeration. I will explain later on, what happened when we do conventional identification. And then plate count methods, the most commonly used methods for probiotic enumeration. We know that it's time-consuming, laborious, highly variable, and non-specific, but still it is a gold standard. Enumeration of specific bacteria requires specialized and standardized methodologies and have not been established for probiotic strain elsewhere in the world. Let's see the identification of probiotic strain. We can do phenotypic or genotypic. With the genotypic PCR and sequencing based on sequence of 16S RNA, DNA sequences encoded 16S RNA for prokaryotes with NCBI+. And then we can do also with the whole genome sequencing. Let's see the function of probiotic, the characterization, sorry. What happened, please? Something wrong. There is something wrong with this. Okay, I'm trying to show you, yeah, that something wrong. Yes. Oh, we start with in vitro, right? In vitro to see whether there is a resistance to gastric juice, to bile salt and bile acid, and an ability to adhere to mucus or human epithelial cells and able to colonize, and then against pathogens, yeah, antimicrobial activities. Then could be also due to displace or exclude or inhibit the addition of pathogens, what we call it direct antagonists on the intestinal cells, no antibiotic transferable resistance gene, yeah, and then continue with in vivo animal experiments, yeah, for example, rats, and in vivo validation with conventional rats, non-SPF or gen-free, able to survive in GI tract of animals. Let's start with my indigenous probiotic lactic acid bacteria. We did the in vitro, sorry. Oh my God. Yes, in vitro probiotic properties, yeah. So the isolated lactic acid bacteria identified conventionally by gram staining, catalase reaction, yeah. We know that this should be conducted with sugar fermentation pattern with a kit, yeah, biomerox kit. We can see with the changes of color, positive or negative, right? And then we did also adhesion properties, yeah, with the human mucus. And then also these two papers, yeah, the research conducted in the laboratory of functional foods Forum University of Turku at Professor Seppo Salminen's laboratory. We did also the cell surface properties, and then we can see here, yeah, that the strain Lactobacillus plantarum IS 10506 showed the highest, yeah, auto-aggregation against pathogens. And then the potential probiotic characteristic also, we did the research and showed that Lactobacillus plantarum IS 10506 also showed the highest addition properties to the mucus. So before we did also the conventional method, right, by identification of catalase, gram, and then carbohydrate fermentation. But this is a very important, I want to let you know. Sometimes, yeah, conventional methods is not quite accurate. When I did the PCR in Professor Seppo, it seems the strain is, the species is not correct. Earlier, we detect as KCI, but then it is Lactobacillus plantarum. That's why we prefer to do the PCR. And then we did also animal experiment. From here, we can see that this probiotic strain IS 10506 showed that at 7 days, consumption, administration of the rats, the viable cell counts of lactic acid bacteria in the faces of rats increased significantly. Even after 3 days wash out period, yeah, still higher than at 0 day. And then also it showed that the strain also decreased the amount of fecal coliform. Another experiment also with bulb C mice. This is Dr. Anang Endarianto is a pediatric who did a PhD in Erlangga University under Prof. Subianto and myself, supervisor. And also comparing the plantarum and Rhamnosus gigi and compared with control. Likewise, the total lactic acid bacteria increased significantly over the time. And we continue with clinical trial. We have two strains of probiotic. One is Enterococcus faecium, IS27526. And we did research to preschool children in Tangerang. And the result here, we can see that as compared to placebo, then the changes of total salivary IgA in undernourished children is significantly higher. And also with the body weight. As compared to placebo, the children supplemented with probiotics, Enterococcus faecium significantly increased the body weight. After 15 weeks. Next is about the microencapsulation. It doesn't mean that microencapsulation can increase all probiotic strain survival because we compare these two. We know this is in vitro, but it is a model of upper GI tract with a computer control, what we call it TIM1. TIM1 is a very sophisticated instrument to check this, which is only available in Canada and in the Netherlands. So with this, we can see that survival of microencapsulated Enterococcus faecium is only 15.7%, when Lactobacillus plantarum 84.5%. So we cannot generalize that all encapsulated is safe, something like that. Okay, talking about horizontally transferred antibiotic resistant gene, particularly those carried within mobile genetic elements and the most likely to be transmitted and thus deserve particular attention. The localization of these genes within the genome, the nucleotide sequence, and the analysis of flanking regions surrounding the antibiotic resistant genes may yield important clues as to the acquisition process of these determinants and their source of origins, as well as their capacity to transfer resistance to other microbes. So we did the whole genome sequencing of Lactobacillus plantarum IS10506 in Maastricht University, the Netherlands. And the result is, fortunately, that this strain contains a single antibiotic resistant gene predicted to confer resistance to tetracycline. Located on the chromosome, we can see the red arrow in the circular graph, and is not in close proximity of any transposable elements in green arrows in the circular graph. Therefore, the chance that it transfer with this resistant gene to any other organisms is extremely low. We can check in these two published papers by Miguel Dumont, 2013, and Lee et al. 2020. Then the sequence is identical to that of several other Lactobacillus plantarum strains, including probiotic plantarum 299V. We can check at this NCBI, and a gene with similar function is also present in best studies of LGG. So the same gene tetracycline resistant found in those strains, but it's not transferable. In conclusion, the strain is safe for human consumption. These are some research published papers, with a strain of Lactobacillus plantarum, starting from in vitro and then in vivo preclinical, and then survival in vitro with computer-based, and also the clinical trials. Okay. With another strain, Enterococcus faecium, we did also the same in vitro and then preclinical trial and the clinical trial as well as the shelf life. I think everybody will have this presentation material. I will not explain all in detail. And then other indigenous strain published papers I show here also with Lactobacillus plantarum .13. Yeah, they published in 3 papers. Let's pay attention to the supplement kesehatan. Yeah. Allow me to say in Indonesian language. Produk yang dimaksudkan untuk melengkapi kebutuhan zat gizi, memelihara, meningkatkan, dan atau memperbaiki fungsi kesehatan, mempunyai nilai gizi dan atau efek fisiologis, mengandung satu atau lebih bahan berupa vitamin, mineral, asam amino, dan atau bahan lain bukan tumbuhan yang dapat dikombinasi dengan tumbuhan. Tujuannya memenuhi dan melengkapi kebutuhan sehingga membantu agar tubuh tetap sehat dan agar tubuh pulih dari kondisi penyakit tertentu. Jadi suplemen kesehatan probiotik itu targetnya mengurangi gejala kesehatan tertentu. Kemudian keunggulan suplemen probiotik, kita bisa menggunakan multi-strain. Kemudian viable counts-nya bisa lebih konsentrat ya, lebih tinggi. Dan kita bisa mengatur bebas alergen misalnya terhadap dairy product ataupun soy, wheat, gluten, and free sugar free or sweetener. The supplement could be in the form of powder, capsule, tablet, or caplet. But we have to prevent to consume different type of probiotic supplement in parallel. Yeah. So the important thing with the food and supplement of probiotic, yeah, there should be label, stability testing, and storage condition. Okay, let me continue. As starter culture in fermented foods, yeah, probiotic can be used as starter culture or can be added, yeah, as a probiotic in processed food, yeah, as a food ingredient. And fiber counts at the end of expired date, colony forming unit per serving should be written, as well as identification of probiotics. Now we talk about enumeration. Okay, probiotic strain could be single or multi-strain, but for multi-strain, each strain should be well identified. And for multi-strain, yeah, multi-strain, it should be written total colony forming unit per serving of the multi-strain, because we cannot differentiate each of the strain for the total viable count. Food and drink, the food ingredient, processed foods or fermented foods containing probiotics. The consume with other, the difference with the supplement, of course, in the food, yeah, we consume the probiotic with other nutrients contents and bioactive compounds available in the food. Okay, so for enumeration method, still gold standard, yeah, for using conventional method, even though we know labor consuming, time consuming, yeah, but still we rely on the conventional method, plate counting, yeah. But so far, no single culture medium is able to carry in an accurate manner for all probiotic products and fails to count viable cells that are dormant or viable but non-culturable. Sometimes the probiotic is still viable, but we cannot grow them properly on the plate because they are in the P, B, and C form. These are the table showing example of standard method for probiotic enumeration. Yeah, you can access the web page, yeah, but then of course based on IDF and ISO. And issue on enumeration of probiotics, yeah, we have no available standardized culture media, need a recommended procedure, and then for multi-strain, it should be in total colony forming unit. Then by definition, probiotic is live microorganisms, yeah, in colony forming unit or live cells. But very difficult right now to quantify different species of multi-species or strain cocktail products. Yes, we can identify by PCR for the existence, but to assess the quantity or level of each strain is hard, even though by flow cytometry. So we know we have flow cytometry, but very sophisticated, and then the cost of instrument and reagents is also something. So need for a specialist to manage the technique, yet still makes them unsuitable for day-to-day routine quality control in many laboratories around the world, especially in developing countries. And with flow cytometry, you get somewhat higher due to analytical difficulties with matrix particle interference. The standardized and accurate procedures for reactivation of dehydration culture, yeah, also for different enumeration, isolation, identification, and for estimation of shelf life, ensuring the probiotic remains viable.
Professor Ingrid,
yeah.
I'm sorry for the interruption.
Yeah.
You only have 10 minutes
Yes.
left. Thank you.
Thank you. So it's hard for us to have standardized protocol, but rather than a part, you can see here if it is single strain or multi-strain. Yeah. So we have to choose the possible culture, the most suitable culture media. And also when we have a frozen or freeze-dried culture, how to prepare it also, it's an important issue. So safety assessment, yeah, safe and no side effect, no systemic infection, no metabolic disorder, overstimulation or immune to vulnerable groups should be prevented, and then no transfer gene, no hemolysis, no delactic production, or should we pay attention on this, especially for children, and free from contaminant. Supporting data for probiotic safety by GRAS, yeah, generally recognized as safe, And then phase 1 clinical trial, no adverse effect and adverse event during the clinical trial, and it is dose dependent. This is the example of hemolysis and antibiotic resistant. But then safety, yeah, non-pathogenic, no toxic production, GRAS, KPS, whole genome sequencing, and clinical trial. And then acid and bile tolerance, ability to colonize, GI tract, stable, viability enduring processing, transfer, and storage. And then safety evaluation of probiotic culture in humans, assessment of adverse effect during clinical studies should be pay attention. And an epidemiological surveillance for undesirable incidents among consumers after has been launched to market. Well, probiotic is alive in the product at an efficacious dose throughout the shelf life. This is very important for the authenticity. Yeah. Yeah. And then talking about strain deposit, should be deposited in national or international culture collections such as INARAC. Yeah, that Indonesia Culture Collection in LIPI, and then VNCC, Food and Nutrition Culture Collection at Gadjah Mada University, and in IPBCC, Institute Pertanian Bogor Culture Collection. And manufacturing or production process also should be well declared to be POM, of course, yeah. And then clinical trial, priority and randomized double-blind placebo-controlled trial, that we have to know the efficacy and the safety of the probiotic, right? We can check the quality of stool and then qualitative, quantitative assessment for microbial in the stool, and then the short-chain fatty acid. Well, the claim for probiotic is the probiotic term itself, yeah, and then maintain the gut health, and then help in maintaining the gut health or some other health issue related to probiotic. So it should be based on the clinical trial, and the confirmation of clinical trial claim in Indonesia, yeah, we can still use from Malaysia, Thailand, Vietnam for Indonesian claim, because then the way of living is quite similar, yeah. The consumption and then nutrient, I mean the eating habits also, and then the hygiene practice also quite similar. Then this is the maintaining the gut health, yeah, because then the microbes can produce nutrients and then provide immune response, positive immune response, help the digestion, yeah, and then producing short-chain fatty acid and also energy harvest. The authenticity of probiotics, yeah, should be in the label. Authentic probiotic food or supplements, yeah, should have a genus, species, strain, and then the amount of viable counts per serving, per milli or per milligram, and also should be remain viable at the end of shelf life. Yeah, so it should be written mengandung probiotic genus, species, strain, something. Colony forming unit per saji, for example. Yeah, and then claim probiotic can be also accompanied by maintaining the gut health or help in maintaining the gut health. So the label, yeah, quality, safety, and function of probiotic should be provided. Well, this is the method for bifidobacteria. I think you can see later on, yeah, this is the, we can compare here, yeah, the different results of agar media showed also different enumeration of viable counts, yeah. So important points for enumeration of bifidobacteria, of course, the diluent, yeah, And then initial suspension, diluent technique, inoculation, and incubation is an important issue. The FAOW Working Group 2002, the probiotic genus, species, name, viable counts at expiration date on probiotics labels. We have to know the availability of accurate, reliable, and fast method for strain identification and viable counts determination, and would facilitate and streamline the process of probiotic authentication. Standard method for probiotic enumeration, plate count method is a gold standard. And then sample serially diluted, plated on suitable growth media, incubated under appropriate growth condition. Special growth media may require anaerobic incubation, for example, and the growing colonies enumerated to determine the viable count as colony forming unit. Well, we can do also the flow cytometry. We can differentiate with live, dead, and also PBNC microbes. We do also DNA based by using real-time PCR or digital PCR, and the health benefits of probiotic are strain specific, dose dependent, and authenticity of the strain as well as viable cells throughout the product of shelf life is very important. Okay, then a key factor to support market growth is therefore the ability to have an accurate count of viable cells in any product. But as yet, there is no single culture medium or combination of culture media that can be used to enumerate all generic probiotics. This is the table of physical states of probiotic strain. Can be in viable or culturable. They can be cultured, replicating, or non-replicating in stationary phase or in hospitable condition or injured. They can be in starving condition or dormant, viable but not culturable, and irreparable damage cell, and some of them may die in a dead state. This is the recommendation for storage and preparation of storage cultures, like storage and then thawing, homogenization, and refreezing. Okay, we talk about the comparison, between among different methods of identification and enumeration. You can see here YFIS, flow cytometry, right? And then PCR. Sir, something wrong. And of course, the cost also is another issue. Okay, talking about probiotic, it is, when we see it's like a sexy girl, the growing market, we can see in Asian market, China is still Asia's probiotic powerhouse. But India and Southeast Asia next to watch Growth Asia Series expert panel. And we can see also Asian probiotic dietary supplement market, small but emerging, right? And this is the probiotic dietary supplement market size, from 2009 to 2014. We can see the growing of the probiotic, right? It's 8%, right? And also, let's see what's up in Europe. I think Prof. Seppo will explain more. Relaxation of the use of probiotic on labeling as Italy, Spain start to use it. Currently, Netherlands also allowed to use it as a previously did. European Commission and EFSA continue with the same view on probiotics. And this country in green do allow the term probiotics on label. Spain, Italy, Greece, Bulgaria, Czech Republic, Poland, and what else? The other one. We can see here Malta. Okay, that's all for today from me. Thank you so much for your kind attention. Thank you.
Thank you, Professor Ingrid. Next, we moving on to our second topic, regulation overview, research and development of probiotic product in Australia with Dr. Patricia Conway. Before we listen to Dr. Conway, I will give you brief information about her. Dr. Patricia Conway, PhD, MSc, is a visiting professor at Nanyang Technological University, Singapore, adjunct professor at the University of New South Wales, Australia, and chief scientist and founder of PCBiome Singapore. She has successfully combined basic research and translational application for over 30 years by being affiliated with the university for supervision of doctoral students and the basic research, while being industry employed, or by working closely with the industry while being university employed. Patricia research interests are gastrointestinal microbiology, probiotic and prebiotic, with the particular emphasis on bacterial colonization and function with the GI tract. She has focused on mechanism of bacterial addition, pathogen inhibition, and immune modulation in animals and human, and the impact of the gut microbiota on health. In particular, health condition linked to inflammation, infection, metabolic disorder, and developing intervention strategies for improving health, especially in infant and elderly. Without further ado, I invite Dr. Patricia Conway. Please, Dr. Patricia, your time is 30 minutes. The floor is yours. Thank you.
Thank you very much for that introduction and the opportunity to participate in this very important topic. And I really have enjoyed the presentations to date. So my overview is an overview today of research development and probiotic products in Australia. So I've limited to what's happening in Australia and try to give you an overview, like a smorgasbord buffet. So if I could have the next slide, please. So I've structured it such that I'll discuss the regulatory aspects first in relation to probiotic products in Australia, and then give you an overview of the type of research that's happening, both preclinical and clinical studies in Australia, and the development of new probiotic strains, and finish with how we're getting the message out to inform the health professionals and the consumers about probiotics and how to use them. Next slide, please. So regulatory aspects of probiotics. The big question, and this has already been raised, are probiotic products food or medicine? We have FSAN, as we call it, which is the food standards for Australia and New Zealand, and we have the TGA, which is the Therapeutic Goods Administration within the Australian Government Department of Health. The big difference really between the two is foods do not have to disclose the number of probiotic bacteria in a product, whereas a product that goes through the TGA, complementary medicines, must show the amount of an active ingredient on the product. So let's now, next slide please, discuss the TGA. TGA, and really the top 2 points listed here are those relevant for probiotics. So therapeutic goods, in the TGA's opinion, are used for humans in connection with preventing, diagnosing, curing, or alleviating a disease, ailment, defect, or injury, and products that are influencing, inhibiting, or modifying a physiological process. So any products that lie within this category would raise the attention of TGA and in most cases need to go via the TGA. Next slide, please. So within Australia, Australia has developed a risk-based approach with a two-tier system for regulating of medicines, including complementary medicines. So there's what we call the low-risk medicines, which can be listed on the Registry of Therapeutic Goods, and the high-risk medicines, which must be registered on the TGA list, on the ARTG list. All listed medicines may only contain low-risk ingredients, and there is a list of what ingredients are permitted, and they must be produced under good management practice guidelines and principles. So even though a listed is a lower risk, it still needs to have the quality consistent with the registered products. Next slide, please. So let's delve a little deeper into that listed and registered. So if it's a listed product with the TGA, it gets an AUSTL, or more recently, an AUSTL in brackets A, referring to that it's a listed product. And registered products are therefore given the registration category, and it depends on the ingredients and the claims for that medicine. So listed products, the companies must hold evidence of the efficacy consistent with their claims, but they are not assessed by TGA. In 2018, a new category was developed, and it's called assessed listed medicines. And these medicines have allowed companies to have intermediate level indications, which must be supported by high quality scientific evidence. Such evidence is not, if it's solely animal studies or traditional use or anecdotal data, those types of products would not be accepted as assessed listed medicines, but rather can still be under listed medicines. So basically, assessed listed medicines that are entered into the ARTG, it's the company's responsibility for self-certification of the safety and quality of the product, but TGA pre-marketing assesses the efficacy of the evidence, supporting the proposed indications. So with just listed, there was no validation or assessment by TGA. With assessed listed, you get the confirmation that TGA has assessed the evidence of efficacy. Could I have the next slide, please? Could I have the next slide? Yeah. Okay, what are the benefits of this new system? Well, largely, it's very effective in that it bridges a significant gap that exists between the evidence required and the costs and the time frame that distinguish between listed and registered. It provides flexibility for the sponsors to access higher level indications and claims that are currently only appropriate for listed medicines without the data requirements, because there are huge costs and the risks of developing the requirements for the registered product. It provides a market advantage to sponsors via the high-level indications. And it also provides significant benefits to consumers and health professionals because of transparency of evidence of claims and the improved confidence in the products. Next slide, please. In addition to the staff of TGA, there is an advisory committee for complementary medicines, and it's called the ACCM. And this is to provide expert advice to the Minister or the Secretary of Health on the safety, efficacy, and manufacturing quality of the complementary medicines. So it was formed back in 2010, and it replaced the previous committee, which was called CMEC. And membership of ACCM is from professionals such as the scientific community, medical, clinical expertise, to guide and give advice to the TGA committee dealing with each application. Could I have next slide, please? Next slide, please. Thank you. TGA also aligns itself to international guidelines. So there's close alignment with the regulatory approaches globally. These include the EU guidelines, which we'll hear from shortly from SEPO, and guidelines compliant with the International Conference of Harmonization, or the ICH, and also the FDA. And the FDA there is the US FDA, although I recognize FDA is also being used by within Indonesia. Next slide, please. TGA also seeks feedback from the community. So this is just an example that I took from the website of TGA, which was a recent seeking feedback on an application to alter international scientific guidelines. And so it's more to share with you that the TGA is very open to feedback, and sure, you need to keep an eye on the TGA website. But there are 370 current adopted scientific international guidelines from the TGA based on this process. Next slide, please. So TGA has recognized that there is an interface between food and medicine, and it refers to it as the food-medicine interface. Generally, TGA sees it that if a product is covered by a food standard, which is the FSAN, or it is traditionally used as a food in the form that it is being marketed, the product is regulated as a food. Otherwise, it is likely to be considered as a therapeutic good. So the TGA has developed a food medicine interface guidance tool, FMIGT, in consultation with the Food Standards of Australia and New Zealand Authority to assist potential customers, and they're very much targeting the SMEs that are new to the system. Big companies not necessarily needing as much guidance, but the SMEs and their first product out. This tool is very helpful for the companies to identify where it should be. Next slide, please. So I will not go through this in detail, but it's more, this is part of the tool that the TGA have prepared for the food medicine interface. So it provides you answer the questions that are in grey, and I've got those on the next slide. And depending on the answer you give as to whether it flows in. So these are the questions that are posed. Question 1, is the product for oral use? Because clearly then it can be food or medicine. And then questions 2 and 3, it depends on the declaration of the product as to whether it's going to be shunted as a therapeutic good or not a therapeutic good. And if it's not the therapeutic good, then the next one is it relevant to a standard under the Food Standards Code? So, for example, if there is a food standard that's directly applicable to that particular type of product, it's clearly a food, not a therapeutic. Is the product a traditional food in Australia or New Zealand in its present form? So it limits the application to Australia and New Zealand, but if it's a traditional food, it is therefore not a therapeutic. And question 6, if the product is represented or likely to be taken for therapeutic use, then clearly it needs to go to the TGA. And so the last one, is the product a class of good that is solely or principal use, or is ordinary a therapeutic use? So it's based on the use and how it's traditionally used. Next slide, please. And this slide is to show you that TGA also has flexibility, because at times we are needing to use product that is not compliant or is not already listed, assessed, or registered as a product. But you can apply to TGA. And examples of when you're needing to use unapproved medicines is the use in clinical trials. How would we be doing clinical trials with products that were not yet categorized with TGA if this system wasn't in place? So there's different applications for approved medicines, and there's a document available, and I'm happy to provide the follow-up information to anybody who's interested. But these are the examples of how TGA will provide approval to use unapproved medicines for these various schemes. Next slide, please. So TGA has a statement on probiotics. They're recognizing that probiotic products, some will come under their umbrella and some not. So as we've already heard the definition of a probiotic, it's live bacteria orally dosed. They see that probiotics are positioned at that food medicine interface. So here I'll discuss how TGA views probiotics to be regulated as foods. As I mentioned earlier, if they're covered by a food standard or traditionally used as a food in Australia and New Zealand, then it's regulated food. Yogurt and fermented yogurt drinks come under the food standard. Yogurt and drinks would be as a food because there is a food standard for it. And accompanying general level health claims for such products can be linked to, for example, lactose health and related claims under that food standard. High-level health claims need to be separately approved by FSAN. So a probiotic product not covered by a food standard and not traditionally used in Australia and New Zealand doesn't automatically end up as a therapeutic good. But the bottom line is, if a probiotic product to be regulated as a food rather than therapeutic, one needs to consider how to market and present it to the consumers, because you can consider your product regulatory-wise as a food, but if there are doses and claims that are more targeting it being a therapeutic good, it therefore does not fall under the food category. So there has to be a close relationship between the marketing team and what's being presented to the consumers to identify whether it is a food or a therapeutic good. Next slide, please. So TGA's application for probiotics as therapeutic goods. So they would view probiotics needing to go through the TGA if there is a therapeutic claim, statement, or instructions for use that would suggest it is to be used as a therapeutic. If it's, for example, if it's to be conceived by the consumer to be used as a therapeutic, even if you intend to market the product as a food. For example, a sachet of probiotic powder may be marketed as a food, but other factors, such as the claims about its health benefits, warning statements, or dosage, may lead the consumer to believe it's a therapeutic good. So this is why it's very important, the working together with the marketing team. So marketing a probiotic as a therapeutic good, then it is regulated as one. Such products are often regulated as the complementary medicines, as we've already discussed. But it's very important, the advertising and the low-level therapeutic codes that are made to be in line with the code. Next slide. So let's now move over to the food standards for Australia and New Zealand and how if your product ends up as a food, how to deal with that. So health claims, it's always the question, and they're in relation to, it's a relationship between the food and the health claim, but they must be, FSAN sees it that a health claim must be supported by scientific evidence. And a health claim is only permitted on a food if it is compliant with the nutrient profiling score criteria. In other words, you can't add probiotic to a food that's very high in fats or sugars or salt and get away with it as a healthy product, because it has to be compliant with the nutrient profiling score to be allowed to have a health claim on it. There are two levels of health claims within FSAN. There's general and high level. General level health claims are often related to a nutrient or substance in the food and its effect on health. For example, calcium is good for bones and teeth. There are 200 pre-approved food health relationships in the food standard, and the FSAN must be notified of the general level health claims that's being used on the food product, and they are to be self-substantiated. So there's to be scientific reviews. The companies need to have a scientific review that's self-substantiated for any health claims that are made. There are also high-level health claims allowed on foods, but to date, there are no probiotic products that have high-level health claims. There are currently 13 pre-approved food health relationships linked to claims, but there are no probiotics in the high-level health claims. Next slide, please. So there's a document which is available on the TGA, on the FSAN website, presenting the relationship between the food and health and the general level health claims. Next slide, please. So as I mentioned, FSAN needs to be notified if you're making a claim. And there's a standard for it, 1.27. And this is an example of the 367 notifications on the FSAN website, of which 72 are probiotic notifications. So I'm not going to show you all 72, but I've put these together to show you that it's the company that submits it. They need to present the ingredient, its claim, and the company details. And as you can see, in one particular case, they have defined down to strain designation and the claim that they're making. The second one listed there simply says probiotic. And the bottom one also specifies the strain designation. Could I have the next slide? And it's more extracts from that list. And I put this together because this particular company has presented as independent claims. Each claim has a separate notification to FSAN for exactly the same product. And then the same, and then at the bottom, it's not it's a food product or beverage, which can have the coagulants and quoting the CFU per gram or per serve, whereas the other products don't. Next slide. And another interesting part of that list of notifications is that a range of companies have identical claims. And so this is for baker's yeast, the beta-glucan. And as you can see on the left, the definition of the product is identical. The claim is identical, but the company is different. So a company can't just take beta-glucan, claim these, without notifying FSAN. So each product and company has to have a notification to FSAN before they can put a claim on their product. Next slide. So FSAN also has an expert group that is assisting them with guidance on what they should do. Next slide. So let's move on to research, and I'm not delving into depth with this, but rather giving you an overview. And when I looked at what's happening in Australia, there's very little development on new strains, but rather utilisation of existent commercial strains. Next slide, please. So I have chose to take strain development from our own work as an example of preclinical studies, and we've used an L-fermentum PC1, and we've carried out, just as Ingrid presented so beautifully in the previous presentation, survival in stomach acidity is crucially important. Our PC1 strain survived extremely well compared to a bunch of other strains. And I will comment that our in vitro survival in acidity, the method we use is consistent with that I used a long time ago, where I compared the laboratory method with in vivo survival in the stomach of humans via a clinical study to validate this method. We've also shown pathogen inhibition. And on the right, you see a box where we carried out a study in mice. First of all, the top two are the growth inhibition of pathogens, And this is particular a salmonella study where we compared the PC1 with an acidophilus. There was significantly more in pathogen inhibition of the salmonella by the PC1 strain compared to the acidophilus. Adhesion, it was much more adhesive to intestinal mucosa. And in the mouse study, in mice challenged with the salmonella, there was weight loss in the mice receiving the acidophilus, whereas those receiving the PC1 had a weight gain, indication that the inhibition of the salmonella by the fermentum was happening in the mice studies. Next slide, please. So I won't go into full details of everything the PC1 strain has done. We've done a mixture of preclinical and clinical studies. I've just mentioned the preclinical, a number of clinical studies, and we're ongoing with mechanistic studies to identify, if we can understand the mechanism behind the probiotic effects, we can propose applications and thereby next slide, please. Next slide. Thank you. So then I've gone into the registry of clinical studies within the TGA within Australia. And these studies, and I've put in bold the topic, just to show you that there's a broad range of topics that are being explored with probiotics in clinical studies currently in Australia. Could I have the next slide, please? So the next one is completed trials. And once again, I've just in bold highlighted the topic. So there's allergic rhinitis, weight measurement, weight maintenance, egg allergy, vaginal infections, depression, cystic fibrosis, pregnancy, resistant depression, hypercholesterolemia, metabolic syndrome, gestational diabetes, antibiotic-associated diarrhea in geriatrics, and sepsis in premature infants. So there's a broad range, and as I said, I think in Australia, probiotic work is focusing on clinical studies. Could I have next slide, please? And so a couple of those studies that have been completed, I've listed here, and I will flag, I have no affiliations with these teams, so it's simply as the publications. So there's the one publication where they're specifying using a mixture of acidophilus and bifidobacterium, and they give the strain designation, and the impact on night shift on immune system. And they showed that participants taking the placebo had changes in the immune system, suggesting a greater stress response than those receiving the probiotic. And it's known as an over-responsive immune system can be deleterious to health. Another study, and this study was coming out of Griffith University, the night shift, and the hay fever study coming out of Griffith University in Queensland. The study is probiotics and hay fever treatment, and it was suggesting that modulation of the immune system was good for the management of allergic diseases. A study coming out of my university is looking at cystic fibrosis, and this publication is the systematic Cochrane review, which was the lead into the clinical study of the application of probiotics for cystic fibrosis. Next slide, please. So let's move now into neonates, because that's a very sensitive group and there is a real need for probiotics, but there's also a need for caution, because there have been evidences of probiotics inducing sepsis in neonates, as listed there. But with careful use, as Ingrid brilliantly presented previously, with appropriate safety and selection of strains, you can safely use probiotics for neonates. And probiotics are used routinely in neonatal units, including intensive care units at 3 of the major hospitals, one in Perth, North Shore in Sydney, and Nepean Hospital in Western Sydney. And I've got the key PI doctors listed there. Next slide, please. So I've worked quite closely with the Perth group, which is led by Sanjay Patoli, at the King Edward Memorial Hospital, but also Princess Margaret. They're associated with the University of Western Australia. This team initially presented and doing the background work of systematic reviews to assess safety, and I've listed there a number of the reviews. The most recent one was 2018, and this review then led into the work we have subsequently completed. The conclusion from that review was probiotics significantly reduce necrotizing enterocolitis via beneficial effects on immunity, inflammation, tissue injury, gut barrier, and intestinal dysbiosis. Next slide, please. So based on that 2018 review, Gayatri has completed a study now, and that's the first one listed there, on comparing a single strain versus a multiple strain probiotic on extremely preterm infants, and showed there wasn't a lot of difference between the two, but safety aspects and clinical benefits have been highlighted in that study. Another of the Perth colleagues, Shripado, has carried out a study with neonates for gastrointestinal surgical conditions. And the second one listed there, it's the application of probiotics in a small group in a pilot study. And subsequently, we've written a paper for the guidelines for future research on the application of probiotics for these gastrointestinal surgical conditions. Next slide, please. So now let's look at, and I have no affiliation with these companies, I just thought it interest to flag. I mentioned peanut allergy as a clinical study earlier. This is now a translational example, because using a commercial probiotic together with an oral immunotherapy, there was benefit shown in infants with peanut allergy. That has now flowed into a company called Protar to commercialize. At the moment, Protar has no commercial products, but they are translating the initial basic research carried out in Melbourne at the university. Next slide. So now there is a probiotic company in Australia called Probiotics Australia, and I have no affiliations with them. They are growing probiotics for commercial sale, as a lot of the major overseas companies. They have the traditional probiotics, Lactobacillus bifido, Streptococcus, Bacillus, Saccharomyces, Enterococcus. They're developing what our first speaker mentioned today, the next generation probiotics. But there are certain regulations that need to be addressed with respect to these next-gen probiotics before they can be commercialized. And this company is looking at 3 of the major next-generation probiotics and developing ways of commercializing, but as yet none of these are commercially available. Next slide, please. So let's just move on to education, because education is a really important part so that we stop just the general word, probiotics will fix allergy, probiotics will do this, rather this particular probiotic will do this. And so there is a health ed program in Australia for doctors, and this company is particularly effective at having educational seminars and webcasts for GPs or anybody who wishes to become a member. Nestlé Nutrition Institute and Danone Nutrition are extremely good at having health professional seminars to increase the level of knowledge and awareness, because if the doctors can understand how to use the probiotic, then they're being used correctly. ILSI is being a major player in trying to get the message across from the doctors, from the researchers to the users of probiotic products. And there's continuing education program for doctors and pharmacists. And the Australian Journal of Pharmacy has published how to use probiotic articles to try to educate the end user and the health professional. Next slide, please. So Choice is a not-for-profit organisation in Australia that is looking across the board at all products that are available. So they also have published their comments on probiotics, and this is at the consumer level. And they flag especially that the number of probiotic bacteria should be maintained at the end of shelf life for a health benefit, and this has been flagged already. Choice also believes that health claims on food or complementary medicines should undergo pre-market assessment by the regulators. Remember, that's not required for probiotic listed products through the TGA. Next slide, please. So I'd like to thank you by quoting one of your eminent professors in Indonesia, Mudani, who says, probiotics and the microbiome are medicines of the future. I think that's where we're heading. Thank you.
Thank you very much, Dr. Patricia Conway, for your very useful insight in regulatory in the Australia and also for the latest clinical trial on probiotic. We are moving on to the discussion session. For first panelist, I will invite Ibu Dwiana to give feedback, comment, or question to our speakers. But as we waiting Prof. Ingrid to enter the room again, maybe Ibu Dwiana can ask for the Dr. Patricia first. Please, Ibu Dwiana.
Okay. Thank you, Patricia, for a very interesting presentation about the regulation, research, and development of probiotic product in Australia. I have a short question for you. As we know, there are variations in the gut microflora among population and ethnics. If there are products from outside Australia that want to be marketed in Australia, is it possible to use safety and efficacy data from clinical studies conducted outside Australia? Or should the manufacturer carry out clinical trials with Australian subjects? Thank you.
Thank you, Bu Dwiana.
Question, and a very relevant question. At the moment, the regulatory authorities are not adding that aspect of the microbiome and the difference between ethnicity. And in some ways, Australia is an incredibly multicultural country. So clinical studies carried out within Australia do in some ways reflect what would happen in almost any population globally. But if it's the other way around, if the study is carried out, for example, in Finland, if Seppo Salminen carries out a study in Finland, how relevant is that in Australia? At the moment, that is not an issue that's being addressed, and it's not, Australia does not at the moment require a study to be carried out in Australia to be considered for regulatory purposes.
Thank you very much, Dr. Conway. And Bu Diana, next maybe you have a question for Dr. Ingrid. Welcome back, Dr. Ingrid, in the discussion session. Ibu Diana may have a question for you, please.
Terima kasih. Pertanyaan Bu Ingrid akan saya sampaikan dalam bahasa Indonesia. Terima kasih Bu Ingrid untuk presentasinya yang sangat menarik. Tadi saya melihat ada beberapa riset yang sudah dilakukan. Saya ingin menanyakan kami dari sisi regulator tentunya apa tantangan dalam pengembangan probiotik untuk penelitian produk komersial di Indonesia. Kemudian selain itu tentu ada tantangan dan ada hambatan. Nah, kira-kira dukungan atau fasilitasi regulasi apa yang dibutuhkan dari kami, dari pemerintah untuk bisa mendorong pengembangan probiotik ini tanpa mengabaikan aspek keamanan dan kemanfaatannya tentunya. Karena kita masih melihat sangat minim ya produk yang bisa diproduksi dan dikembangkan lokal, jadi kami berharap ini semakin banyak. Dan mungkin ada pandangan atau ada masukan kira-kira dukungan atau fasilitasi dari sisi regulasi apa saja yang dibutuhkan untuk mendorong penelitian ini. Terima kasih Bu Ingrid, Prof. Ingrid.
Boleh langsung.
Terima kasih Ibu, silakan Bu Ingrid.
Terima kasih pertanyaannya Ibu Diana. Sebelumnya, allow me to say hello to Professor Patricia. Hello. Patricia, nice to see you virtually.
Lovely to see you again, Ingrid.
Yeah.
Nice to see your work.
Yeah. Saya akan menjawab pertanyaan Ibu Diana mengenai tantangan ya, challenge. tadi sebetulnya di presentasi saya kan sudah disampaikan bahwa probiotik ini seksi loh, ya kan? Benar-benar apa namanya? Pasti banyak dilirik gitu karena itu challenge ya, dalam artian Indonesia ini membutuhkan khususnya kalau probiotik itu secara umum lebih banyak yang memberikan efek, memberikan respon imun, meningkatkan respon imun. Padahal kita tahu kita banyak sekali kan kendala-kendala atau masalah-masalah kesehatan, apalagi selama COVID-19 kemarin gitu. Nah, kemudian itu sebenarnya kan sebagai tantangan ya, kita harusnya punya produk yang memang benar-benar unggul karena dia seksi, maka harus dijaga benar oleh Badan POM kan gitu. Jangan sampai ada misleading bahwa memang kalau probiotik itu authenticity-nya memang guaranteed gitu ya. Strain specific, dose dependent, efficacious begitu. Nah, untuk kalau ditanya hambatan, saya sendiri selama ini mungkin belum terlalu jelas gitu regulasi yang untuk probiotik. Tapi sekarang semakin mengerucut gitu ya, semakin ada kejelasan begitu. Dan menurut saya sih apa yang menjadi requirement dari regulatory itu bisa semua dibuktikan begitu. Kalau diminta in vitronya ada, in vivonya ada, clinical trialnya ada, safety assessment ada, efficacious dose ada. Kenapa tadi saya presentasikan tuh sebagai model lah ya, bahwa strain saya tuh ternyata dari in vitro sampai clinical trial sampai safety sampai whole genome sequencing bisa dibuktikan dan memang doable gitu ya kalau diminta. Itu sih dari saya. Terima kasih.
Thank you, Professor Ingrid. And for the next panelist, I invite Bapak Raymond Chandrawinata. I haven't mentioned before, Bapak Raymond Chandrawinata, PhD, is the Director of Business Development and Scientific Affairs of PT Dexa Medica. Please, Bapak Raymond, your comment or question.
Okay. Thank you very much for your for the opportunity to give comments. I really enjoy the talks by Professor Ingrid from Venus University, and also Professor Patricia Conway from the UNSW from Australia. I would like to give some comments and suggestions in light of the Bangga Buatan Indonesia movement, obviously is commanded by our President Bapak Jokowi recently, which definitely we need to increase the numbers of Indonesian discovered and developed products, including, obviously, the topic that of our heart today is probiotics. And also recognizing the recent progress by Kementerian Kesehatan, Department of Health, in issuing formularium for phytopharmaca recently, as recent as last week, for our phytopharmaceutical product for Indonesia that have gone through to clinical trials. So Indonesia, in this light, as alluded by the speakers, we must have several usages of probiotics. But again, we have to recognize the fact that a lot of probiotics in Indonesia, in this country, are still, many are being legally registered in Indonesia, but only few, like for example, there are only 53 products as supplements and only 4 products as food. So we need to increase the number of probiotics in this country, obviously. But with the 50, the limited number of probiotics, Indonesia currently depends on probiotics which is imported into this country. Even some are not legally registered by Badan POM. Yeah. So if we gather the marketing authorization data from the website of Badan POM, there are 52 out of 53 supplements product, which is 98% imported into this country. There's only one product which is locally made, it's called Lakbon, produced by Kimia Pharma. And in the category of food products, 3 out of 4 products, which is 75%, are imported there's only one local product, which is Yakult, that we recognize from, again, the Badan POM side. So obviously, we need to increase the number of probiotics which is produced, even discovered, and also developed in this country. We also have heard from Professor Conway earlier presentation, in other countries such as in our neighbor Australia, the Australian TGA or even the other organization facilitate the process of registering supplements product with indications for health maintenance, that is for general health, obviously. Where the product category is included in listed medicine, means no need pre-market assessment, not as registered medicine. So it should be common goal to reduce number of imported probiotics products to accelerate the availability and production of probiotics developed domestically with probiotics isolates originating from Indonesia. In that regard, there is a dire need for support of regulatory agility by Badan POM, emphasizing a risk-based approach, which is alluded by Professor Conway just earlier, which we also heard, to increase the number of domestically produced probiotic products. For example, we would like to suggest the clinical trial requirements are only required on new strains that are genetically and functionally different from the strains already registered. Another one, for new strains that have genetic similarity to strains that have been registered previously, i.e. known from the, for example, identification through the whole genome sequencing, the industry applicant must be allowed to apply for a waiver not to do clinical trial, as long as the indication follow the indications of the strain that has been previously registered by Badan POM. Another suggestions, strains that are safe to use, that is actually obviously on the Badan POM positive list, can be expanded not only to strains that have been registered with Badan POM, but also include strains that have the GRAS classification, generally recognized as safe, or also included in the QPS qualified presumption of safety list of EFSA, or maybe also having supporting data from international reference journals that also contain the safety study. So somebody already done the study, and we can use as a reference for that matter. Yeah. For the strains that have the GRAS classification, generally regarded as safe, we assume that there will be no need for toxicity tests or clinical trials because they're already listed in the GRAS documents, as long as the indications or claims are still relevant, because it's already been GRAS, generally recognized as safe. We also would like to suggest that the probiotic strains isolated from sources in the form of food, for example, from the breast milk, fruit, and etc., can also be considered as safe probiotics because the originating is actually human, for example, the breast milk or fruit and so on. So those are our suggestions from the discussion that we have heard, the beautiful presentation from Professor Ingrid, Professor Patricia Conway, and we would like to suggest these suggestions, and hopefully we will have more probiotics discovered and developed locally in Indonesia, which make us proud, and that will actually obviously will fill up the gap that we have for the Bangga Buatan Indonesia, as commended by President Jokowi. Thank you very much.
Thank you very much, Bapak Raymond. I think we all agree that we need to increase local probiotic product from Indonesian industry. So let's work together between Indonesian FDA, industry, and also academia to support that program. So Bapak Raymond, there's no question from Bapak Raymond to our speaker. So I will moving to next panelist. She is Dr. Agnes Febrita. She is the General Manager of Kalbe Pharma. Please, Dr. Agnes, the time is yours.
Thank you. Pertanyaan pertama untuk Ibu Ingrid. Ibu tadi selalu menyebut dose dependent dan adequate amount of probiotic ya Bu. Nah, sementara dalam proses pembuatan, dalam manufacturing itu sering kali dilakukan overage ya, supaya pada saat dikemas sampai dengan expired date-nya itu dia masih mempunyai jumlah viable cell-nya dipertahankan seperti itu. Nah, apakah ada formula khusus untuk menghitung overage dari si viable cell-nya ini, Bu?
Formula khusus untuk apa, Bu?
Untuk overage, untuk menjamin si viable cell-nya ini tetap ada dengan jumlah yang adekuat dari awal sampai dengan akhir masa expired.
Oh, terima kasih Bu pertanyaannya. Boleh langsung dijawab?
Silakan Ibu.
Jadi kalau memproteksi kan ya sebetulnya. Memproteksi itu formula khusus bisa dengan mikroenkapsulasi, tapi tadi saya sudah memberikan gambaran ya bahwa itu bukan berarti jaminan bahwa kalau sudah di mikroenkapsulasi dia memang survivalnya bisa dipertahankan. Harus ada penelitiannya Bu. Jadi bisa dengan pakai, kalau misalnya kalau kebetulan saya punya kolaborator yang punya alatnya ya, yaitu dengan timuan. Jadi kelihatan memang survivalnya itu bisa ketahuan kalau itu di dalam GI tract. Tapi kalau untuk viable count untuk expired date ya sebagaimana layaknya semua apapun makanan, minuman ataupun suplemen ataupun obat kan ada expired date-nya juga pasti ditentukannya dari penelitian kan Bu ya. Jadi kayak seperti itu. Jadi harus dengan uji begitu. Apakah dengan accelerated test atau half life atau dengan yang real time gitu. Seperti itu Bu menurut saya. Karena nggak bisa kita mengeneralisir gimana caranya supaya kalau dia di suhu ruang dia viability-nya bertahan gitu ya. Seperti itu. Terima kasih.
Thank you. Do you have a follow-up question, Dokter?
For Patricia, yeah, I think the question is still the same. Yeah, it is about the adequate amount because the probiotic we know it is dose dependent. Yeah. What do you think if in the manufacturing process the industry make the overage of the viable cell, let's say 10 times of the doses at the manufacture date until the expiry date to guarantee the quantity still the same. And when the patient or we take the product with the overage number of viable cell, it is fine for the consumer to take the dose with more viable count.
It's a very good question and a very important topic to discuss. Yes, clinical studies are carried out at a particular dose. But remember, the dose that's used in that clinical study is also influenced by the storage conditions of the probiotic. So a clinical study is supplied with the capsules at the start of the clinical study and then stored as instructed. But that product is, for example, 6 months old before the subjects are eating it. So the clinical study is not absolute. Each patient is exactly the important strains and also the stability studies carried out by the manufacturers. And for example, in our neonatal studies in Perth, I was very stringent on the probiotics that were being used in those babies, had demonstrated stability studies at the storage conditions that were being used for the life of the product, not just at the start of the product, but at the end of the product. So and as Ingrid has mentioned, the quality of the product and the quality of the data from the suppliers is crucially important. We know that 10 times numbers of bacteria is not a big difference. It's if it's dollars, it's a big difference. but 10 times bacterial numbers doesn't make a lot of difference in terms of question.
I think
if a higher dose is being used, is it if a lower dose is being used? It's just come off the production line, has more organisms. Can you hear me?
I'm sorry, Dr. Patricia, but I think we have a technical difficulty.
Yes, I could see that.
Yeah, we cannot hear some of your answer. Maybe you can repeat, but before that, maybe Dr. Agnes will try to emphasize which one to be repeated. Thank you.
Yeah, maybe you could repeat after 10 times dosage.
Yeah, or basic, I'll simplify my answer is probably a good way of doing it. Yes, a probiotic product at the start of production has high numbers and at the end of shelf life has lower numbers. So is there a risk that at the start of shelf life, the product is not safe because that was not the dose used in the clinical study? The other aspect, at the end of the stability, the numbers are lower. Will it be as efficacious? But the clinical study was subjected to the product used in the clinical studies is effectively under the same parameters as the product. So the clinical study was supplied with product at a certain count and subsequently used over 6 months. So the clinical study was carried out using a variable number. I would flag that some strains are very stable. Some strains die off rather quickly, but it is also dependent on the production. So I have produced a probiotic product in a capsule, which is stable at room temperature, but conditions used for the production influence how stable it is. So it's very much supplier dependent, but in terms of safety and efficacy, which I think are the important things, 10 times more is not at risk of being unsafe. And if there's a drop, it may be not as efficacious if the product is not stable. But the good suppliers of probiotic product supply the end of shelf life stability data for specific storage conditions.
Thank you very much, Dr. Conway. Is
Boleh
that
saya
clear,
tambahkan,
Dr.?
Bu?
Yes, please. I think Prof. Ingrid want to share some comments, please.
Tadi Dr. Prof. Patricia already explained, right? Then it's true that we base on the efficacious dose, right? So by the end of expired date, it should be at the efficacious dose, the viable counts. That is the important point. But then how to make it stable and everything, it depends on the strain itself, right? And also if you increase the dose, of course, you should also have the safety assessment data up to how many viable counts means it's proven safe, something like that, right? Thank you.
Thank you, Ingrid.
Thank you for both speakers. Thank you.
Thank you very much for both speakers. Maybe Dr. Conway have the final remarks before we are closing this discussion session. Please, Dr. Conway.
I would just like to say it's such a pleasure that very important issues have been raised during the morning session and the before coffee and this session. And I think probiotics are a must in the future, but it's really important that we have safety considerations and efficacy considerations. And sadly, there are very many probiotic products on the market now that are claiming those loose health claims and have multiple strains, none of which have had any evidence of efficacy. So I would be very supportive to assist Indonesia, to the health authorities, to ensure that products called probiotics actually have strains with some evidence of efficacy. And it's sad when dodgy products are scattering the market and being lumped together with the quality products.
Yes, thank you very much. It's very true what you shared about there are many products in market that we need to try to understand whether they have valid evidence base. Okay, and next maybe final remarks from Professor Ingrid, please. Do you have final remarks?
Yeah, thank you so much. Yes, we believe that this hybrid meeting will go towards the final regulatory for especially for this supplement division at BPOM, as well as of course the foods division. And then I believe that with the final regulatory being issued, then it will encourage the industry, especially the local industry, to start with their products. Probably some of them are still waiting. Thank you.
Thank you very much. Ladies and gentlemen, we are reaching to the end of the discussion. Thank you very much for all speakers, Dr. Conway and Professor Ingrid, for the meaningful and valuable insight about probiotics in Indonesia and Australia. And also, I would like to say thank you to all our panelists, Ibu Duyana, Bapak Raymond, and also Dr. Agnes. Once again, on behalf of Indonesian FDA and all participants, we would like to say thank you. Thank you very much. I will give the time to the MC. Thank you.
Thank you very much for the speakers, panelists, and moderator. Distinguished ladies and gentlemen, now we will have a lunch break for about 15 minutes. After lunch break, we wish to all participants to come back in this platform at 13 for Jakarta time. Thank you.
Pegangnya gara-gara pada mundur gitu kali ya sesinya.
Indonesia is the most beautiful land in the universe. The land where we were born, the nation where we grew up. So many colors that live together in peace. With a million timeless treasures, with an abundance of natural wealth. Indonesia is not just wonderful. Indonesia is a wonderland.
Kota baru gunungnya Bamega, manapun di Salangkara, manapun di Salangkara. Batamu lawanlah Adinda, Adinda iman di dadara, Samalayang iman di dadara, Samalayang. Soleram, 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 anak yang manis, anak manis, janganlah dicium, sayang kalau dicium, merah pipinya. Kampunan jauh di Mato, Kunwangsan sebaku cobi nangis anakku sing ayu rupane tetiap pendekare bangsa.
Proklamasi kami bangsa Indonesia dengan ini menyatakan kemerdekaan Indonesia. Hal-hal yang mengenai pemindahan kekuasaan dan lain-lain diselenggarakan dengan cara bersama dan dalam tempo yang sesingkat-singkatnya. Jakarta, 17 Agustus 1945, atas nama bangsa Indonesia, Soekarno, Hatta.
Indonesia is the most beautiful land in the universe. The land where we were born, the nation where we grew up. So many colors that live together in peace. With 1,000,000 timeless treasures. With an abundance of natural wealth. Indonesia is not just wonderful. Indonesia is a wonderland.
Kota baru gunungnya Bamega, Bamega, Bamanapur di Salakara, Bamanapur di Salakara. Batamu lawanlah Adinda, Adinda iman bidadara, Samalayang iman bidadara, Samalayang. Sayang, sayang, si patokaan, matego, tego, gorokan. Sayang, 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 si patokaan, matego, tego, gorokan. Sayang, sajojo, sajojo, yumadam ko bisa berpare samuna muna muna keke, samuna muna muna keke. Soleram, 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 anak yang manis, janganlah dicium, sayang kalau dicium, merah pipinya. Kampuanan jauh di Mato, Kunwangsan saya baku liliang, takkan aja kawan-kawanan lamo, sangke basuh yang surya.
Indonesia dengan ini menyatakan kemerdekaan Indonesia hal-hal yang mengenai pemindahan kekuasaan dan lain-lain diselenggarakan dengan cara yang sama dan dalam tempo yang sesingkat-singkatnya. Jakarta, 17 Agustus 1945, atas nama bangsa Indonesia, Soekarno, Hatta.
Paling manuk cingklang cing datang, loskan kolom bapak satan bulanan. Mendaki jalan pandai sike, menurun jalan kebiaro, bima hati indah mampe, awan kaki cuan, oi oi ayam belape. Layang-layang, selayang pandang, hati di dalam, bagai bergoncang. Jangan ragu dan jangan bimbang, ini lagu selayang pandang. Pukul gandang garam tuh, hayat dengan kangkalu, sambil tuntang gang tahu, biar mundur dan maju. Ayo menari manasai, menari manasai, menari manasai. Belenggang patah-patah, nganape goyang pica-pica, nganape body bocok-bocok. Tenggang-tenggang lopi na anak koda ipanjaja buluana. Pinang muda dibelah dua, gunung kerinci, gunung yang tinggi menjulang. Wahai anak muda, kalau berjalan harus berdua, senanglah hati walau tiada orang yang larang. Mana di mana anak kambing sayang, anak kambing tuan ada di pohon waru. Mana di mana jantung hati sayang, jantung hati tuan ada di kampung baru. Cacang marica hei hei, cacang marica hei hei, cacang marica ada di kampung baru. Cacang marica hei hei, cacang marica hei hei, cacang marica ada di kampung baru. Ayah mama, jangan mama rabeta, dia cuma pegang beta. Ayah mama, jangan mama rabeta, lah orang muda punya biasa, lah orang muda punya biasa, lah orang muda punya biasa. putarlah ke kiri, ke kiri, ke kiri, ke kiri dan ke kiri, ke kiri, ke kiri, ke kiri manis. Sekarang kanan, mudah manis, putarlah ke kanan, ke kanan, ke kanan, ke kanan dan ke kanan, ke kanan, ke kanan, ke kanan manis. Loloskan kolom bapak satan bulan.
Welcome back to all distinguished participants, ladies and gentlemen. No further ado, the topic and next session is regulatory overview, research and development of probiotic product in Finland and Southeast Asia countries with the speakers Professor Seppo Salminen, MSc, MS, PhD, and Dr. E. Seong T. The session will be facilitated by the moderator, Ms. Fitri. Over to you.
All right. Thank you very much, MC. Good afternoon, ladies and gentlemen, the honorable guest speakers, and the honorable panelists, and also all the participants. I'm very pleased today to see you here and welcome all of you to the third and last session of International Scientific Probiotic Research and Potential Development of Probiotic Products for Health and Regulatory Study of Probiotic in some countries. Ladies and gentlemen, there will be two speakers in this session. First speaker is Professor Seppo Salminen, MSc, MS, PhD from Finland. And second speaker is Dr. E. Seong T from Malaysia. Before the presentation begin, please allow me to inform how the presentation will be going on. Firstly, Professor Seppo Salminen will be invited to present the material for 30 minutes, followed by Dr. E. Seong T to present for another 30 minutes. After that, there will be question and answer session by the panelists for around 40 minutes. If they still have time, we will take questions from the audience. All right. For the first speaker, Professor Seppo Salminen, are you here with us?
Yes.
Next, okay, hello. Next to me, there's Prof. Ingrid. I want to say hi to you.
Hello, Prof. Seppo Salminen. Thank you so much for being with us, even though I know you are in a very difficult situation. You are hospitalized. Get well soon and speedy recovery. Thank you so much.
All right. So I would like to introduce Professor Seppo. So Professor Seppo is the president of the International Scientific Association of Probiotics and Prebiotics in Finland. Good morning in Finland, Professor.
Good morning.
Hello, good morning. So now is your time to deliver your presentation. The floor is yours.
Okay, can I please have the screen sharing? Okay. Good morning from Europe, ladies and gentlemen. I guess it's afternoon for you. I will present the ideas of probiotics and health claims, safety and novel foods as they are done in Europe by EFSA assessment procedures, because in Finland we are part of the European Union and we continue to follow the regulations that are set by the European Union. And in terms of probiotics, it is the European Food Safety Authority, EFSA, that assesses both the safety and the health properties. I thought that I would take first this important slide to show you the difference between, for instance, the American assessment procedure and the European assessment procedure. In Europe, the European Food Safety Authority is a scientific risk assessment and risk communication body. So EFSA does not make any final decisions. The final decisions are always made by European Commission, European Parliament, and EFSA acts as their scientific support organization. So EFSA does scientific risk assessment, scientific advice and risk communication, but the risk management is all reserved to the European Commission. Inside the European Food Safety Authority, we have about 550 officers today working on different parts of food safety. And there are several panel, expert panels, and the probiotic work is part of the NDA panel, Nutrition, Dietetics, and Allergies panel. It has usually around 20 to 25 scientific members and several working groups. And the important thing is to remember that although we consider probiotics and other biotics important, it's just one part of the NDA panel to work on these. The discussion in Europe has very much focused on claims, health claims, and you know the situation that there's actually only one claim that's related to health claim that's related to probiotics, and that's yogurt and alleviation of lactose intolerance symptoms. But other than that, the focus on claims regulation has been very challenging for probiotic researchers. If we think about the focus, really, the basic idea is consumer protection. The claim regulation and probiotic regulation is meant to avoid false or misleading claims, and that means that claims must be scientifically substantiated. The other part of the regulation is to improve the free movement of goods within the European market and harmonizing the rules between different countries. That means that if in one of the European Union countries something is accepted, it should be accepted in other countries, member states as well. And the third idea of this claim regulation has been to promote innovation. And it has certainly promoted a lot of research in probiotic and prebiotic area, but it's not quite sure if it's promoting innovation. It's promoting agreeing to the regulations and following the book of the regulations. So consumer protection is the most important. So it means that claims and probiotic claims as well should be understandable by an average European consumer. And they should be not misleading, and they should be scientifically evaluated by EFSA. One has to remember here that claim regulation is not meant for healthcare professional communication. This means that medical societies, for instance, can communicate claim regulations and claim recommendations within their own area. And that kind of communication is meant for healthcare professionals, and it's not regulated by the health claim regulation itself. So what is a health claim or a nutrition claim in Europe? It's interesting and important to remember that any presentation or description where there is a statement or a picture, and that the picture or statement leads consumer to believe that the food has specific health properties, that is considered a health claim. It can be text on the package, it can be pictures, or it can be symbols. And usually health claims are messages which are not obligatory by other types of regulation. So it's something we will voluntarily add after the scientific assessment to the health claims. There's an important part which is difficult to find sometimes, why we don't see claims on probiotics in Europe or prebiotics. And it is the Standing Committee on Food Chain and Animal Health in European Union that has decided that a health claim is a health claim if the naming of a substance is related to an indication of function or implied effect on health. And this means that, for example, contains antioxidants. The function of antioxidants is a health claim, is not permitted. And it has also led to the idea that the word probiotic and prebiotic, the name contains already a health claim. So if you want to use the probiotic or prebiotic name, you should have EFSA assessment behind you. And that is an important factor, and that's why we don't see too many references to the probiotic word in food area. but we do see a lot of mentions of probiotic in healthcare professional communication, including for instance, pediatric societies and their recommendations for probiotic use for preventing or even treating diarrhea. So that kind of communication is allowed because it's meant for health professionals only and not for the common consumers. What else is there important in the regulation assessment? And the important factor one is that main claims are meant for general population, healthy population. They are not necessarily at all meant for people who have different diseases, and they should be based on generally accepted science. Sometimes it's difficult, it's challenging to understand what is meant by generally accepted science, but it comes out with the scientific assessment of proposed claims, because we have to look at the causality, the so-called cause-effect relationship, and we have to conduct a scientific assessment, as it says on the claim regulation, of scientific assessment of the highest standard. And transparency in all these actions, all assessments and the background documents are published in EFSA Journal. EFSA Journal is an open access journal that's available by Wiley Publishing Company, and it lists all the EFSA assessments that have been conducted in full. So what are the important criteria for substantiation of a health effect? First of all, in the EFSA panel, the general agreement is important, so that everybody agrees with the claim regulation and suggested claim. But we have to consider that the most important factor starts from characterizing the food and characterizing the effect. Is it plausible, and is it important for human health, and is it beneficial? And at the end, all assessments are done by weighing all the evidence available. The criteria for a health claim include a cause-effect relationship. So if we do give a probiotic dose, is there a cause-effect relationship in the host? And is it feasible in the normal food and diet? If we, for instance, require 2 liters of kefir for obtaining a health effect, it's not feasible in the normal food diet. Another criterion is also that the claim is scientifically established and defined for a target population. The target population could be vegans, vegetarians, normal consumers, or different groups like that. And that target population is required to be defined in the EFSA assessment. I tried to take a couple of examples, but as we don't have them from probiotic area, I thought that I will take an easy example: plant sterols. There are several plant sterols in the market in Europe and globally, and there are several food matrices that are applied to these products. And compounds have been tested in several food matrices, and even in the beginning, novel food safety assessment of plant sterols was done by EFSA. So it's quite an important example because it has undergone novel food safety assessment and claims regulation, health claim assessment. There are several products. I listed the products and food matrices in the black here, compounds tested in several food matrices, and novel food assessment conducted and approved by EFSA. And in many studies, long-term and short-term studies, the components were well defined. The basis for the effects was sufficiently characterized. Clinical studies in several countries, several EU member states, and product assessment in target populations, by meaning populations with high cholesterol or slightly elevated cholesterol. And at the end, in most of these studies, a cause-effect relationship has been demonstrated. And therefore, the health claims have been approved, sometimes modified. For instance, for plant sterols, the NDA panel on European Food Safety Authority established a statement saying that blood sterols have been shown to reduce blood cholesterol, and blood cholesterol lowering may reduce the risk of coronary heart disease. So that was the end of the scientific evaluation. Thereafter, as we started with in the beginning, the responsibility for making the decision went to the EU Commission, and the EU Commission slightly changed the suggested health claim. So at the end, plant sterols have been shown to reduce blood cholesterol, and high cholesterol is a risk factor in the development of coronary heart disease. So that was finally the wording that was approved by the Commission. They also wanted to say that plant sterols should be consumed only by people who need and want to lower their blood cholesterol, and that patients on cholesterol-lowering medication should only consume the products under medical supervision. So you see that the process is quite long. It requires several steps, and even in case of plant sterols, after the first approval, there have been several pre-applications for different food groups. Another example could be a probiotic drink. And by the way, as said, these examples are defined in detail in the EFSA journal. So if you go for asking questioning, for instance, for Evolus product, all the details are there. But Evolus was a fermented probiotic drink with the active peptides, and the producers wanted to propose a claim reduction of arterial stiffness, saying that Evolus has been shown to reduce arterial stiffness and consequently lower the risk of the cardiovascular disease. And these products were low lactose or no lactose products with specific peptide composition. However, the claim was done in methods that were not generally accepted. So the proposed claim reduction of arterial stiffness caused a lot of discussion. And it was concluded that it has not been established that reducing arterial stiffness is a beneficial factor for human health. And it has not been demonstrated that it reduces the risk of coronary heart disease or cardiovascular disease. And therefore, a cause-effect relationship was not established, and the claim was not approved. So it's important to remember that whether the claim has been approved or whether it has not been approved, still all the scientific assessment is in a transparent manner in the EFSA journal by Wiley publishers. And everything is open access, available to anybody who is interested in it. The factor that has raised a lot of discussion has been what is the health effect? Because health claims can be only given to components that reduce the risk of a certain disease or reduce the risk of certain discomforts in food system. And no claims can be given on prevention of disease or treatment of disease. So we have to look at the prevention as a factor for drugs and treatment as a factor for drugs. But we could say that a food can be reducing the risk of disease if certain risk factors have moved into the favorable direction. And I took here as an example a potential prebiotic, xylitol, 5-carbon polyalcohol. It is one of the most important things because it presents a factor which is present in most of us. 98% of the people have caries. So it's a factor that's related to normal population, and therefore reducing a risk of caries is certainly a benefit. And this was one of the first claims that was accepted. Xylitol and tooth decay, xylitol chewing gum and pastilles were proposed for the reduction of risk of tooth decay. And the chewing gum was sweetened with 100% xylitol, no other sweeteners, and pastilles with 56% xylitol. And the claimed effect was reduces the risk of tooth decay. And the target population, as said, we all have caries of varying degrees. So the target population is really the whole European population in this case. So again, we start with accurately describing and characterizing the product. So it was in this case very easy. Chewing gum, only xylitol as a sweetener. And the important thing in chewing gum studies was that we had biomarkers. We had clear biomarkers. The biomarkers were thickness of dental plaque, number of oral streptococci, the DMF index, decayed missing teeth and their amount, and the postprandial pH change in saliva, indicating actually that if there are a lot of oral streptococci in your saliva, they will ferment glucose very fast, sucrose very fast, and produce a change in pH to the lower pH values, and that could be detrimental for dental decay. But in this case, all these factors move to the right direction, and the cause-effect relationship was established in terms of the chewing gum, but not with the pastilles. So the biomarkers of risk of tooth decay in children and later on in adults, they all changed into a favorable direction. So thereafter, the health claim was issued. Xylitol chewing gum reduces the risk of caries in children, later on also in other populations. And the quantity of chewing gum sweetened with 100% xylitol can be consumed as about part of balanced diet. However, it was noted that chewing gum should be avoided by children less than 3 years old because of potential challenges in choking, for instance, if the chewing gum is not discarded. And at the end, this was one of the first health claims, and the commission wording is like this: chewing gum sweetened with 100% xylitol has been shown to reduce the dental plaque, and high content of dental plaque is a risk factor for development of caries in children. Hereafter, also other non-sugar chewing gums have received health claims, just as also artificially sweetened chewing gums have received health claims. So the health claim situation is quite difficult, and we don't really have any probiotic-related health claims approved. However, there are health claims for prebiotics, so the carbohydrate components that promote healthy gut microbiota, and most of those are condensed on areas like improved function, maybe improved bowel function, reducing the risk of diarrhea, or improving improving the constipation symptoms. So there are possibilities, and we have perhaps we have to carefully consider how to promote probiotic studies so that they actually correspond to the targets of EU regulation. Another factor that I wanted to take here at the end was the safety assessment. We have seen an emergence of quite a number of new potential probiotics that are not of the traditional food fermentation bacteria. And here we have the assessment of novel foods regulation in Europe. However, the novel food regulation can be avoided if your microbe belongs to the so-called Qualified Presumption of Safety QPS database. This is a database that has been established by the EFSA Biohazard Panel. And it's basically conducting safety assessment of microbes and other biologicals that may be used in foods or animal feeds. Again, the QPS database is evolving, but the most updated ones is always available in the EFSA journal. And the important thing is that it lists microbes and the background data for microbes which are considered safe in foods. Another important factor is that it's updated annually. And of course, if there is some kind of a problem or cause for alert, a rapid assessment can be updated. You all know that assessing microbes for safety is quite important, and it's also quite difficult. How to assess the safety of a live microbe, because live microbes can always, under certain circumstances, cause a problem, even an infection. This has been also noted by probiotic researchers, and the novel food requirements are quite strict in Europe. So therefore, proving the safety of a live microbe is quite challenging. And therefore, live microbes are often inanimate, meaning that they are not live. We don't usually say inactivated because we want to have inanimate microbes with health effects. And we are seeing more and more of those in the European market and assessment all the time. Okay, I think I'm stuck now. Okay, so novel microbes, novel probiotics in European Union regulations are often postbiotic or inanimate microbial preparations because the safety assessment for live microbes is so challenging. If you have the possibility of selecting a potential probiotic that belongs to the QPS system, it makes life a lot easier. The regulatory assessment is easier for non-viable microbes, and it differs quite a lot from the US GRAS regulation and GRAS notification system. Key examples of the differences in novel food microbes and postbiotics are shown here. The first one, Clostridium botulicum, caused a lot of concern when it was submitted for safety evaluation because Clostridium botulicum had never been used in foods in European Union countries. So therefore, that required novel food assessment. And after a long-term assessment and comments from European member states, the Clostridium botulicum was accepted as safe, but there is no health claim for Clostridium botulicum in Europe at the moment. The first inactivated or inanimate preparation that came into the safety assessment was Bacteroides xylanisolvens. Novel food assessment does not include any health benefits or any health requirement. It's only assessing safety. So Bacteroides xylanisolvens was assessed as a bacterium that could be used for fermenting mild milk products and related products. And at the end of the fermentation, the safety assessment requires Bacteroides xylanisolvens to be inactivated by heat treatment, and it even defines the temperature and time that is required for the inactivation to make the microbes inanimate. So that was the first product, inanimate product. The second one, the last, was Akkermansia mucinifila. You may all know about it. Akkermansia is one of the few microbes that has been demonstrated to have beneficial health effects, both in the live and in the inactive form. So the safety assessment of Akkermansia was just done during the last year, and it was accepted as safe, and therefore it can be brought into the European market as a dietary supplement, for instance, but not yet with health claims. Maybe there will be more healthcare professional communication again here. So these three top bacteria, Clostridium botulicum, Bacteroides xylanisolvens, and Akkermansia mucinifila, were approved as safe, and the Commission reinforced that. Then EFSA panel analyzed and did safety assessment also for Mycobacterium cetens, and it was concluded safe even in 14-day dosages, but the EU Commission has not authorized the use because they have considered that food should be safe in all doses, and no restrictions should be given to the use of food components in the long term. So quite an important group of bacteria. And then potential future applications. There are several bacteria that are being tested in live and in inactivated form, listed here, for instance, like Fecalibacterium prausnitzi, fructophilic lactic acid bacteria that can be found in honey and mead and in fruits, and several others. And it seems that most of these bacteria are actually submitted for safety evaluation in the inanimate form, again to ease the process of risk assessment and evaluation of the safety. At the end, I wanted to take this thought of the International Scientific Association of Probiotics and Prebiotics. We have now 4 types of prebiotics that have been set for a consensus definition: probiotics, prebiotics, symbiotics, and postbiotics. And there's also a consensus document on fermented foods. All these consensus definitions have been published in Nature reviews in gastroenterology and hepatology, and they are open access. The idea here has been to provide a common basis for global assessment so that we would go through and find a way of defining the biotics, defining the QPS system, defining health claims in a similar manner in different circumstances and globally. So this has been done only so far by yogurt, for instance, has an international standard that is accepted by most countries around the world, and it makes it easier for assessing then the health benefits and also assessing the safety and future potential of fermented foods with different microbes. So as the postbiotic definition is the most recent one, I took it here as an example, the preparation of inanimate microorganisms and or their components that confers a health benefit to the target host. So trying to get an example, this postbiotic publication in Nature Reviews is perhaps the most recent and best one to demonstrate that we have got the experts from different areas globally and professionally and combine them together to reach a consensus. And this may perhaps offer new ways of continuing also the regulatory assessments in the future. Thank you very much.
All right, thank you, Professor Seppo, for giving us a very informative and interesting presentation. We greatly appreciate it. We wish you a speed recovery, and thank you once again for joining us and sharing with us today. The next speaker to present is Dr. T. Yi Seong. Are you here with us, Dr. T?
Hi, Madam Chair. Yes, I was not able to unmute myself. Yes, I'm here. Thank you.
All right. Yes, so allow me to share his profile. Dr. T. Yi Seong is the president of the Nutrition Society of Malaysia and chair of the Southeast Asia Public Health Nutrition Network. He is also a co-chair of the Southeast Asia Scientific and Regulatory Expert Network. Dr. T is also scientific director of the International Life Sciences Institute, or ILSI, Southeast Asia region, and also coordinator for Malaysia Country Committee. So without further ado, I would like to invite Dr. Yi Seong T to deliver his presentation. And Dr. T, the floor is yours.
Thank you very much, Madam Chair. My audio okay? Okay. Thank you, Madam Chair. I would like to thank Badan POM for giving me this opportunity to participate in this important scientific webinar where multi-stakeholders are present, and including regulatory agencies, food industry even, and also academia. So allow me to share my slides. My slides appear okay?
Yes. Would you like to full screen? Yes, thank you very much.
Okay, thank you. Thank you very much. And I have been requested to speak on regulatory overview of probiotics products in Southeast Asia countries, meaning how do Southeast Asia countries regulate products containing probiotics in foods and beverages, as well as in supplements. Sorry. Okay. It's not moving. Okay, sorry. So this is my presentation outline. Just 2 slides very quickly on the increasing demand for probiotics and the science for scientific evidence for probiotics. Then the focus of my talk will be on a survey that we conducted, and I will highlight 6 topics from that survey, Number one is the regulatory framework. How do countries regulate probiotic products and legal definition for probiotics? Secondly, what are the permitted microorganisms used in these countries for use as probiotics in food as well as in health supplements? Then third topic is how do companies apply for new probiotics to be used within the 6 countries in our survey? And fourth topic is, are there any permitted function claims or health claims? And what are these claims? Number 5 is, are there any specific labeling requirements? And what are these labeling requirements? And finally, the last topic, I will also share with the participants some other regulatory control with beyond Southeast Asia region, in Asia, in Europe, and in the USA. And finally, Madam Chair, 2 slides on my concluding thoughts on what's the way forward for the regulatory control of probiotic products in the Southeast Asia region. So I did not go into detail on this. I think Professor Ingrid also mentioned about increase of probiotics, and there's a lot of science now, scientific substantiation of the health benefits of probiotics from many research groups all over the world. Yeah? And especially in the area of digestive tract, immune system, and respiratory functions. So these are also increasingly promoted or advised recommended by healthcare professionals. So number and type of foods and drinks containing these products are increased. And I saw in the Google Drive for this webinar, there are various products already in the Indonesian market. Yeah? And so these have become increasingly popular functional foods for use by the consumer. Well, probably because the consumers are becoming more learned, more interested in their health and well-being, and also looking for more healthy products available in the market. And if we look at the last 2 years, the increase in demand for probiotics has increased, yeah? And maybe because of the COVID pandemic, and thinking and hoping that these probiotics will help them improve or strengthen the immune system. So we are aware of this FAO-WHO expert consultation in 2001, which already has definition on probiotics. We have heard other speakers mention in today's webinar. However, after 22 years or 21 years, we'll see that there's no harmonized regulations. We already heard some aspects of this in this morning's webinar. So no harmonized regulation on probiotics, no harmonized industrial practices on probiotics. So this often leads to various concerns and issues and challenges. And this could be challenges for the regulators, could be for the food industry or manufacturers of probiotic products, and even to the consumers. So this relates to the quality of the product. Consumers are not sure. Safety of this product. We also heard about some not so genuine products or confusing products, and also labeling confusion across the different countries. So this is just a quick slide to share with you the increase in the compound growth rate of probiotic products in Asia Pacific region. And this is, I took from Model Intelligence. They also quoted that could be due to the COVID-19 pandemic, the surge in demand has been witnessed in the last 2 years. The Model Intelligence report also highlighted that because of the complexity in the regulation, the legislative framework, and technological complexity of manufacturing of probiotics, then these have become a major hurdle for market growth of probiotics in Asia Pacific region. So I will now go on to talk about, talk about the survey that we conducted. This was a survey that we conducted under the Southeast Asia Probiotic Scientific and Regulatory Experts Network. It was started by Professor Hadinsha Pergizi Pangan, and I was co-chair. I'm still co-chair of this network. It was started in 2019 in Jakarta, and this provides a platform for the various stakeholders to get together and share experiences and and also updates on probiotic scientific and regulatory aspects of probiotic network, all the key stakeholders, much like what Dr. Penny Lokito mentioned, the 5 parties, stakeholder, the regulators, academia, professionals, and also the food industry. So this platform has been we have been meeting firstly in Jakarta, in Surabaya, and after the lockdown, we met online, and we have been meeting 5 times. So one of the activities that we did was we thought it would be useful if we combine all the regulations in the region and compare and contrast the differences among these regulations to understand what's the status, and are there differences, and what are the differences or similarities amongst these regulations. And this review has been published in the Malaysian Journal of Nutrition last year, and it's open access article that we can download from this journal. So I will be sharing highlights from this review article. As I mentioned, 6 topics I'll be sharing, and the first one is the regulatory framework. The regulatory framework, we can note that countries have taken different approaches. All the 6 countries in Indonesia, Malaysia, Philippines, and Thailand, they have specific probiotic regulations. And within each country, maybe 2 different organizations or branches of the organization take care of different products, different meaning foods and beverages and health supplements. In Indonesia and Malaysia, there are 2 different agencies taking care of the 2 different categories of product. On the other hand, in Philippines and Thailand, both categories of products are taken care of by the Food and Drug Authority, FDA. On the other hand, Singapore and Vietnam do not have a specific probiotic regulation. However, products containing probiotics are permitted to be sold in these 2 countries. So as I mentioned, there is a legal definition of probiotics, and Professor Conway and Professor Salmanen have already alluded to this definition of probiotics. The consultancy was 2001, and then ISAP has defined or refined very minor editorial refinement of the definition in Hill et al. 2014. And this is the definition: live microorganisms that, when administered in adequate amounts, confer health benefit on the host, on a human being, for example. So these are the regulatory agencies that are covered in this survey. Badan POM, 2 different agencies. This is for supplements, the agency organizing this seminar today, and the processed foods. And this is in Malaysia. The supplement or health supplement is controlled by National Pharmaceutical Regulatory Agency, and for food is by the Food Safety and Quality Control Division. In the Philippines, both are by the same agency, FDA. In Thailand also, both are by FDA. These are the countries with clear regulation, specific regulations on probiotics. So this is the FAO-WHO definition I took from the 2001 report, and 3 important aspects we all are aware of. For example, they must be live microorganisms. They must be consumed in adequate amounts. We have heard many times already, and they must be demonstrated to confer health benefit on the host. So this is the ISAP definition 2014 that Professor Hill and his groups, including Professor Salmanen, published in 2014, and the definition is very similar to that of FAO-WHO. So let me go on to the second aspect of our survey. What are the microorganisms permitted for use as probiotics in these 6 countries? And this again differ a lot amongst the 6 countries. And also only a few countries have a positive list. Malaysia has a positive list of what probiotics are permitted to be used. Permitted list means only these probiotics are permitted to be used. Anything not listed here are not allowed to be used. That's how Malaysia controls. And in the case of health supplements, there is no positive list published for the consumer or industry. Philippines and Thailand also have a positive list, but different probiotics are permitted. I will show you the list in a short while. Yeah? And if we look at these probiotics allowed to be used, mostly are from the Bifidobacterium and also Lactobacillus species. On the other hand, Indonesia, Singapore, and Vietnam do not publish a positive list. Yeah? That industry can find out, or consumers can find out what are to be used. Additionally, we look at the regulations. Malaysia and Thailand make it compulsory that probiotic containing products must contain a minimum amount of 10 to the power 6 CFU per mil or per gram of this strain of probiotics. So this is very quickly showing you from the regulations in the Malaysian list. There are 32 strains up to the strain level. This is from the Philippines. Philippines has 5 genera. They're listed in genera. And Thailand has 23 species permitted to be used in this published positive list of permitted probiotics. Yeah? The third topic that I will be sharing with you from the survey is how do companies apply for the use of new probiotic strains, probiotic cultures. Yeah? So all the 6 countries in the survey allow for companies to apply for new probiotics to be used. However, different regulatory framework, different approaches, slightly different requirements. When we look at the survey findings from the regulations, we find that there are some general similarities amongst these 4 countries that have clear regulations. That means Indonesia, Malaysia, Philippines, and Thailand. There are general similarities. For example, they publish guidelines for companies who want to apply for new probiotics. Clear guidelines are given. And identification of probiotic strains, data to show identification of strains must be provided to the regulatory agency. Identity of genus, species, and strain using current valid methodology and nomenclature, much like what Professor Ingrid mentioned just now. Yeah? So data on tests to determine characteristics of the probiotic strain must be provided to the regulatory agency. And data to show ability to survive in the digestive tract, data to show adherence to the mucosal epithelial cells, and ability to hydrolyze bile salts. All this data must be provided. And further on, safety assessment requirements are very similar amongst these 4 countries. For example, ability to determination of antibiotic resistance pattern must be demonstrated. Assessment of metabolic activities, for example, D-lactate production. Assessment of side effects of these cultures must be clearly demonstrated. Even if you do beneficial trials or trials to demonstrate beneficial effect, if any adverse effects are reported, these must be reported to the regulatory agencies. And of course, evidence of safety of combination of strains. If multiple strains are in that product, we have also heard about multiple strains, then evidence must be demonstrated, provided to show that there are no antagonistic effects. In fact, there should be synergistic effects demonstrated. Yeah. So finally, the requirement is that all clinical data to support the beneficial effect must be demonstrated. So remember, we talk about the third effect is that demonstrated beneficial effect to the host. Yeah. And these must clearly demonstrate functional role or health benefits to the host. So these must be demonstrated through well-designed, randomized controlled trials by all the 4 countries that participated in this survey. Yeah? And in addition, in Indonesian Badan POM for the food control requires that these data must be obtained through local studies and conducted on Indonesian populations. This is the requirement by Badan POM, yeah, for the food control. So let me go on to the fourth topic of our survey. That is, were there any pre-approved functional claim or health claim by these 6 countries? If we look at the Malaysia, Philippines, Singapore, or Thailand, they do have a published pre-approved generic, functional, or health claim. Pre-approved means that companies do not need to apply to use these claims. They can just use these generic claims. Yeah. For example, I'll show you these claims later on, but in Indonesia and Vietnam, there's no published pre-approved claims. So these claims, when we look at all the claims and put them together, they are related to intestinal or gut function, or digestive health, or healthy digestive system, or words of that kind of similar meaning, or they refer to intestinal microflora, microorganisms, yeah, microorganisms, or healthy intestinal ecology, environment, that kinds of wording, or refer to desirable balance, or good balance, or healthy balance of good bacteria in the gut. So these are the claims I took from the actual regulations. In Malaysia, only one generic claim: probiotic cultures help in improving intestinal or gut function. In the case of supplement, this is the pre-approved health or function claim. In Philippines, 4 pre-approved claims. In Thailand, this is the pre-approved functional claims, very generic functional claim. In Singapore, for health supplements, there's no pre-approved list, but such claims are permitted. Support, for example, support or promote healthy digestion, or support or promote digestive health. These may be used on the food label, or on the supplements label, sorry. For the food, these are the pre-approved claims: maintain digestive system, help digestion, and these kinds of wordings, such as shown on the screen, are permitted. Yeah. So let me go on to further on the health and functional or health claims. All countries allow for additional function claims to be used, or generic health claims to be used. Yeah? And the companies can apply to this: Malaysia, Philippines, Singapore, or Thailand. They must be, of course, be provided accompanied by scientific evidence obtained from human clinical trials. In the case of Indonesia, for the food related to food claims, this must be obtained through clinical trials, obtained through local studies conducted on Indonesian population. And the regulation 16 or 2016 provides very clear information on how to conduct this clinical trial. In the case of Vietnam, it is not clear what is the requirement for submission of health claims or functional claims to the Vietnam Food Authority. Okay, move on to the 5th topic: specific labeling requirements. Countries have different labeling requirements from these 6 countries that we survey. 5 countries, Indonesia, Malaysia, Philippines, Thailand, and Vietnam, require that labels must clearly identify the genus, the species, and the strain of the probiotic. In addition, a number of live microorganisms must be, towards the end of the shelf life, must be indicated on the label, and storage instructions must be clearly indicated on the label. Singapore has no specific labeling requirements for probiotic containing products. In Thailand and Vietnam, these are required. However, there's also additional requirement that they must carry a cautionary statement. Caution, like, for example, this is for health supplement. Supplement is not a medicine, and this supplement cannot be used as a substitute for medication in case the consumer misuse this health supplement. Okay, this is just an example I took from the Malaysian food regulation to share with you the different components of the requirement. This here, the words containing live probiotic cultures must be given there. And then the culture name must be given there, genus, species, and strain up to strain level. And storage instruction must be given there. And then number and quantity of the product must be stated. For example, 1 billion or 3 billion or 10 million must be given there. And also number of probiotic cultures must not be less than 10 to the power 6 CFU per mil or per gram. So let me go on to the last topic of this survey, and that is going beyond South Asia, what is the status of regulatory requirements? We also heard already from Australia, Professor Conway very clearly explained already, but this is the other countries in the world. And I took this from a report by Au et al. 2019, and she looked at 11 Asian countries, Australia and New Zealand. So there's divergent approaches on how probiotic products are categorized. Some call them general foods, some are under functional foods, some are under nutraceuticals, some under health supplements or foshu, like in Japan, health foods or novel foods. And also different probiotic strains are approved for use in all these 11 Asian countries and Asia Pacific countries, and different permitted claims. And the procedure to apply for new probiotic strains differ amongst these different 11 Asia Pacific countries. Another review I took was from Professor Nomoto, 2021, contained in this monograph, functional food monograph, published by International Life Sciences Institute. That's a chapter on probiotics. And he reviewed the regulations in different continents. In North America, only Canada and Brazil have enacted regulations. And as we hear from Professor Salmanen, there's no specific regulation for the whole of European Union, but some countries do allow. Yeah, Professor Ingrid did share with us just now. In Asia, regulations are there in China, Korea, and India, and no specific regulation in Japan. However, the foshu system does allow claims related to GI tract for lactobacillus and bifidobacterium. Yeah? So across the world, we'll also see different kinds of regulations on probiotic. Yeah? So this is the last bit on Codex Alimentarius. We speak about harmonization of regulations across the regional South Asia or across the globe. So countries found that there is no regulation. We already saw so many surveys indicated that. Yeah? There's also concern that there are products in the market sold as probiotics, but they do not meet the FAO/WHO definition or WHO et al. Definition. Yeah? So countries recognize that there's a need to develop a Codex Alimentarius guideline for probiotics. As we are aware, Codex Alimentarius is international body, international food standard setting body. Yeah? So it is best for Codex Alimentarius to provide guidelines so that they can become consistent eligibility criteria as a guide to countries when they want to establish their own national regulations. So Codex provide these guidelines. Then if more countries follow these guidelines, then there will be more harmonized or similarity in the various probiotic regulations. So these guidelines, these regulations then will meet the needs of authorities, consumer bodies, and also the food industry dealing with probiotics. So if there's harmonized guidelines by Codex Alimentarius, it is believed that it will lead to better consumer protection, better satisfaction, confidence in these products, because there's a global guideline that many countries follow. Yeah? So of course, this harmonized guideline will facilitate international trade, making more products, more choices available to the consumers in different parts of the world. Yeah? So this is the proposal to Codex Alimentarius. Argentina did make a proposal in 2018 at the 40th session of Codex Committee on Nutrition and Foods for Special Dietary Users. So Argentina was asked to refine the proposed harmonized guideline on probiotics. And Argentina did present the revised guideline in 2019 in Germany at the 41st session of Codex ACC NFSDU. In 2019, Argentina was asked to further make improvement by some countries who see that maybe some areas need to be improved of this guideline. So that was done, and during that meeting in 2019, Malaysia offered to work together with Argentina in improving this harmonized or proposed harmonized guideline. So in 2023, Argentina and Malaysia will jointly table this revised guideline, much revised guideline for discussion at the 43rd session of CC and FSTU, hopefully in March in Germany. So just last part on harmonization of regulations in South Asia countries. So under this South Asia Probiotics Scientific and Regulatory Experts Network, we have been discussing on exploring opportunities for harmonization among the ASEAN countries or South Asia countries. Some of these areas include definition, positive list, general function claims, labeling requirements. Maybe these are the areas that could be harmonized across the countries among South Asian countries. Yeah, and this, our network, Probiotics Strand, provide this common, we are discussing providing a common framework or guideline, developing this guideline for the use of countries in the region, regulators in the region, for the evaluation of new probiotic cultures. If such a guideline becomes available, then countries in the region will evaluate every application more or less in a similar manner. A similar requirement can be provided to different countries. Yeah? So this framework or guideline we are developing include data for identification and nomenclature, requirements for characterization of the strains, safety evaluation, including what are the requirements for human clinical trials, what are the requirements for clinical trials to demonstrate functional role or beneficial health effects of the probiotic that the company intends to sell in that country. So, Madam Chair, this is my last slide on my concluding thoughts. Yeah? So we can really predict that the demand for probiotic foods, probiotic supplements will continue to increase in the Asia-Pacific region. So because of that, then, it's very important to have clear regulatory control by the regulatory authorities for the control or the marketing and advertising of these probiotic containing products. So with that clear regulation, then consumers can have access to safe and efficacious products in foods and as well as health supplements. So then also enable the food industry, manufacturing industry to have a clear understanding on the requirements of each country, on what are the new probiotics strains that may be approved by their different countries. And this then provides a level playing field. With a clear regulation, a level playing field can be provided for all industries, and that products that do not meet the guideline, do not meet the definition, should not be on the shelf, so that consumer get genuine probiotics. If they label as probiotics, they must meet the requirement for probiotics. Yeah. So it will be significant benefit if there's harmonized probiotic regulation among the Southeast Asia countries. So with this harmonized regulation, then development of probiotics in the region can continue to improve and progress and expand. Yeah? It will facilitate trading of safe and genuine probiotic products in the region. And of course, this expansion will require capacity building of research and development. They will require regulatory review and development. And of course, a collaboration amongst the different key stakeholders is crucial to enable this harmonization and expansion and R&D to take place. Yeah? So regulatory authorities, these stakeholders would include regulatory authorities, research groups in the region, academia, professional bodies, and also the relevant industries, such as happening in this webinar today. Yeah. So finally, I've also argued that it's very important for consumer education programs to go on at the same time. I hear Professor Conway also alluding to that. So this can be done by the regulatory agencies, by the health authorities, by professional authorities, by professional bodies, by healthcare professionals, yeah, and to advise the consumer what are the appropriate use of probiotics. This is not a savior for everything under the sun, for all the ills under the sun. Also advise the consumer how to choose genuine probiotic containing products. I think this is my last slide. Thank you very much for listening. Thank you, Madam Chair.
All right. Thank you very much, Dr. T, for a very comprehensive presentation. And we would like to explore more through the Q&A session. All right, ladies and gentlemen, now we come to the question and answer session. I would like to introduce our panelists for this session. First, there will be a panelist from Indonesian Food and Drug Authority, or BPOM, Director for Traditional Medicine and Health Supplements Control, Bapak Irwan, SSI APTMKM. And then we also have academician and practitioner from Gajah Mada University, Professor Dr. Insinyur Endang Sutriswati Rahayu MS. And we also, the third panelist dari Ikatan Dokter Indonesia atau Indonesian Medical Association, we have Dr. Slamet Budi Santoso, Master Pendidikan Kedokteran. And the last one, we have from the industry, PT Agromitra Alimentari. Okay, without further ado, I would like to invite Professor Endang. Are you here with us? Professor Endang?
Hello.
Hi. Hi, Professor. All right, so I will give you time to, if you have inquiry or questions to Dr. T, please, the floor is yours.
Yeah. Good afternoon, everybody. Actually, I have a slide that can be presented. Someone?
I think yes. The committee, please do it. Professor Endang could share her screen. Sudah bisa dicoba, Ibu?
Oh, slide dari ini.
Oh ya, dari panitia ya Bu ya, berarti ya yang present ya. Share screen-nya maksudnya.
Okay. Yeah, I would like to explain about the relation between probiotic and gut microbiota. Yeah. The main requirement of probiotic strain is their ability to survive and grow in the digestive tract and convert health benefit, of course, in addition to its safety assessment. So the main mechanism of probiotic strains after being consumed is growing together with the commensal gut microbiota, which about trillions in numbers. Probiotics are expected to be able to maintain the balance of gut microbiota, produce beneficial metabolites, and also provide mucosal protection and other mechanisms which give a health benefit to us. The development of gut microbiota is influenced by various factors such as age, geographic, medicine, and mainly diet as well as lifestyle. Many research and publications show that the gut microbiota mainly influenced by diet and lifestyle. For example, our publication explained that there are 2 enterotypes in Asian children which supported by diet and lifestyles. Indonesian and Thai children have a Prevotella enterotype, which characterized by high consumption of carbohydrate plantation. Meanwhile, in Japan, China, and Taiwan, with their diet high animal protein, the children have Bacteroides enterotype. So it is clear in Asia that there are 2 enterotypes based on the difference of diet. So it is clear. Yeah. I cannot move my cursors. Okay, gut microbiota, it's really supported by what we eat and also our lifestyle. Many publications explain that the imbalance of gut microbiota, or known as dysbiosis, is associated with various diseases. Dysbiosis is not only related to intestinal disorder, but also related to other organ diseases. Like in these pictures showing that now many terms come out in the publication, like the relation between gut microbiota and brain axis, and gut microbiota metabolic disorder, gut microbiota liver axis, and currently related with SARS-CoV-2 virus, gut microbiota with lung axis. There are so many publications correlate between consumption of probiotic to many to other organs, support consumption of probiotic in the purpose to maintain the health of other organs, not only gut, not only intestinal health. So it is clear, the main role of probiotic is not only maintain the balance of gut microbiota, but also maintains the balance atau to restore gut microbiota during dysbiosis. Many articles now showing that the health benefit of probiotic beside intestinal health, such as brain, liver, lung health, and also together with their mechanism. The health benefit of probiotic greatly depend on the use of strain. It means strain level. So I would like to give a summary. First, gut microbiota enterotype really influenced by diet and lifestyle. Beneficial effect of probiotic showed by its ability to integrate with particular gut microbiota for each community based on diet and lifestyle. New probiotic must be supported with proof which demonstrated its ability to integrate with gut microbiota for each community based on their diet and lifestyle. Regulation in Indonesia stated that it's mandatory to conduct clinical trials on Indonesian population. Harmonization of probiotic regulation in Southeast country, such mentioned by Dr. T, is possible to attain because of the similarity diet and lifestyle among the Asian country, as well as identical gut microbiota enterotype, especially in Indonesia and Thailand. So I have no question for Dr. T, but I mentioned here that we support of the harmonization of probiotic regulation in Asian, particularly based on similarity of gut microbiota. So I gave my time back to the moderator.
Thank you, Professor, for
Professor Endang. Thank you, Professor Endang.
Oh, okay. Thank you also, Dr. T. So the next, we would like to invite Bapak Dr. Slamet Sudi Santoso, Bapak dari Ikatan Dokter Indonesia. Silakan Bapak apabila ada tanggapan ataupun ada komentar terhadap presentasinya Dr. T.
Thank you. Good afternoon, Dr. T, and good afternoon, everybody. Introduce me, my name is Dr. Slamet, representative to Indonesia Medical Association. Appreciate and your presentation and nice to meet you, Dr. T. Sorry, I am to use Indonesia speaker. Okay, Dr. T? Yeah.
Okay, I can try to understand. I should be able to.
Ya, Indonesia dan Malaysia satu rumpun ya.
Sama aja.
Ya, Indonesia menjadi satu prinsip utama bahwa Ikatan Dokter Indonesia ini sangat menjunjung tinggi hak asasi kesehatan manusia yang memang harus diwujudkan dalam bentuk pemberian berbagai upaya layanan kesehatan, terutama dalam hal ini adalah suplemen, melalui beberapa penyelenggaraan pembangunan kesehatan yang berkualitas. Sehingga memang dibutuhkan bahwa dalam upaya mewujudkan kesehatan ini yang tadi disampaikan oleh Dr. T, bahwa masyarakat juga harus mampu menjangkau produk-produk yang dibuat menjadi suatu probiotik. Di Indonesia ternyata berbeda dengan negara-negara ASEAN lainnya seperti Malaysia, Singapura, dan sebagainya, bahwa probiotik itu sudah merupakan suatu hal dasar dalam menjaga kebutuhan. Hal ini terkait dengan pemahaman bahwa probiotik itu di Indonesia masih dianggap sebagai minuman kekinian itu apa ya? Modern. Sebagai minuman modern yang tidak dianggap bahwa menjadi suatu kebutuhan rutin sehari-hari, sehingga kita tahu bahwa produk-produk yang dibuat itu memang harusnya memberikan pemahaman di konsumsi setiap hari. Selain memberikan suatu layanan kesehatan, maka untuk memberikan itu juga Ikatan Dokter Indonesia memberikan suatu perlindungan dan kepastian hukum terhadap produk-produk yang diperlukan, sehingga memang perlu ada suatu pengaturan. Tadi juga sudah disampaikan, ada label, ada indikasi, lalu ada komposisi, dan juga ada batas waktu expired. Nah, pengaturan produk-produk ini diharapkan mampu berkualitas, aman untuk dikonsumsi, dan tetap menjunjung tinggi moral etika produk kesehatan. Jadi tidak semata hanya menjual suatu produk. Dan juga perlu menggunakan suatu evidence-based medicine. Selain itu, produk-produk ini juga harus tetap melakukan suatu pembinaan atau pengembangan dalam rangka meningkatkan mutu yang lebih baik. Dan tentunya harus ada monitoring efek samping dari suatu produk yang dihasilkan. Nah, memberikan suatu perlindungan hukum kepada penerima pelayanan kesehatan, perlu ada produk-produk yang memang distribusinya diatur, karena pemahaman masyarakat Indonesia bahwa proses penyimpanan atau proses distribusi yang tidak dipahami begitu baik, sehingga akan merusak produk probiotik yang dihasilkan, karena memang suhu juga mengganggu proses di dalam perkembangan bakteri tersebut, sehingga diperlukanlah di Indonesia diperlukanlah suatu edukasi tentang manfaat probiotik, karena melihat banyaknya manfaat probiotik menjaga kesehatan ini, sehingga jenis-jenis produk probiotik, efikasi dan efek samping, aturan penggunaan, faktor yang harus diperhatikan, ini juga memang harus mendapatkan pemahaman di masyarakat, khususnya di Indonesia, karena melihat dari kemasan, kondisi penyimpanan ini juga menjadi satu hal yang sangat penting. Selain itu, Ikatan Dokter Indonesia juga berpikir bahwa dalam riset pengembangan produk probiotik sebagai mikroba yang menstimulasi pertumbuhan mikroba lainnya, perlu juga dipikirkan apakah perkembangan bakteri-bakteri lainnya yang bersifat patogen ini akan memunculkan, karena sempat terbersit ada suatu efek minimal atau yang tadi sudah disampaikan oleh pembicara sebelumnya terkait dengan infeksi seperti infeksi sistemik, sepsis, atau mungkin kembung. Tapi kami melihat tadi ada beberapa produk-produk yang dibuat yang ternyata bahannya itu di Indonesia tidak import dan dibuat di lokal sendiri. Tadi saya sempat melihat ada 2, dan mudah-mudahan ini akan meningkatkan pengembangan probiotik di Indonesia. Thank you, Dr. T. Terima kasih.
Thank you, Bapak, for your thoughts and very good thoughts you're sharing with us. And the condition is similar in Malaysia. We need a lot of education to the consumers on very basic things like storage, how to use the products properly. Yeah, so they should include it in their daily diet, not take once a month or twice a year. It's not going to be beneficial. Yeah. So thank you for your thoughts. Yeah.
All right, thank you, Dr. T. Thank you, Bapak Slamet. So next, we would like to invite the panelists from industry PT Agromitra Alimentare. Kepada Bapak Apoteker Gerekta Geslan, apakah sudah join bersama kami di sini? Oh, di sini mau. Oh, baik-baik.
Nggak tahu ada acara apa gitu ya, nggak tahu. Oke, kalau gitu saya diskusi dengan ini nanti saya panggil juga ya.
Thank you for Professor Saminen and Dr. Seong Tae for nice and meaningful presentation. My comment and question for both of you. My comment and question in Indonesia, and my son Gary will translate into English. Kita mendapat asupan probiotik secara alami mulai dari kita lahir ya. Kita mendapatkan probiotik itu dari jalan lahir dan minum ASI, breastfeeding ya. Kemudian kita juga mendapat probiotik itu dari air microbiome yang masuk melalui saluran pernafasan dan kontak dengan kulit.
So first of all, in the beginning of our lives, we get our first taste of our probiotics from the mother's passage from natural birth and the consumption of breast milk. Afterwards, we adopt the microbiome and probiotics from breathing the air around us and coming into contact with our surroundings through our skin.
Asupan probiotik lain kita bisa dapatkan melalui makanan-makanan yang fresh ya, khususnya produk-produk perkebunan dan pertanian ya. Pada produk-produk perkebunan dan pertanian seperti pada buah dan sayur-sayuran ya, terdapat probiotik alami. Jadi kalau kita makan fresh food, kita juga akan mendapat probiotik alami yang ada di dalam produk tersebut.
And so fresh food specifically will contain a lot of probiotics as they are harvested from natural farms and grounds. And those probiotics will eventually reach our bodies through the digestive tract.
Di dalam sayur dan buah juga terdapat banyak serat non-digestible fiber yang merupakan makanan dari probiotik yang hidup di dalam usus kita.
And in those fruits and produces are fibers that can be utilized as prebiotics in order to support a healthy microbiome growth in our digestive system and bodies.
Metabolit yang merupakan produk dari soil microbiome ya, itu akan masuk ke dalam sayur dan buah-buahan. Selanjutnya, metabolit itu akan menumbuhkan gut microbiota, sehingga gut microbiota itu akan mirip dengan soil microbiome.
And there might be a chance that the soil microbiomes from the fresh fruits and produces will make it into our bodies and will eventually produce metabolites that will create an environment in our digestive tract that is similar to the soil microbiome.
Dengan demikian kita bisa memiliki komunitas mikroba yang beragam dan seimbang yang dinamakan mikrobiota.
And that is why through the contact of different microbes, we have what is called a gut microbiome.
Lingkungan yang buruk khususnya di perkotaan itu tidak kondusif untuk hidup dan berkembangnya probiotik, sehingga asupan alami itu sudah tidak kita dapatkan.
And especially in urban and city and developed areas, we come less into contact with natural microbes and the metabolites, which will eventually cause a disruption in our natural gut microbiome.
Ditambah lagi lifestyle yang buruk, ya bad lifestyle, orang perkotaan mudah stres, makan makanan yang bergizi tidak seimbang, makanan yang banyak lemak, manis, ya karbohidrat sederhana seperti tepung, ya kurang istirahat karena terlalu banyak aktivitas, semuanya berakibat pada kerusakan mikrobiota yang dinamakan dysbiosis.
And because of that, the phenomena of dysbiosis is caused by unhealthy lifestyles adopted by people living in the city and urban areas. And those factors can be stress, lack of nutritional food intake, and the intake of foods that are detrimental to our health, such as refined sugars and foods that are really high in carbs and fats.
Dysbiosis menyebabkan terjadinya keradangan kronis, ya. Penelitian terkini membuktikan bahwa semua penyakit relate dengan keradangan kronis, baik itu penyakit kanker, penyakit kronis seperti gastritis, diabetes type 2, ya, hypertension, and infection also.
And recently, there might have been a link between these conditions with the emergence of diseases and ailments such as cancer, diabetes, gastritis, and etc.
Tujuan suplementasi probiotik, ya, adalah untuk membangun mikrobiota yang beragam dan seimbang, eubiosis, ya. Dysbiosis direstorasi menjadi eubiosis.
So eubiosis is the result of the restoration of a gut that has been affected by dysbiosis, and eventually by consuming these beneficial probiotics, we will restore the gut by diversifying the amount of the types of microbes, and so that there will be a healthy composition inside our guts.
Metabolit dari probiotik akan menumbuhkan bakteri lain yang menguntungkan, sebaliknya akan menekan yang merugikan. Dengan demikian akan terbangun mikrobiota yang beragam dan seimbang.
A balanced and diverse gut microbiome can be built by consuming probiotics, which will eventually support the growth of beneficial microbes while also suppressing the growth of pathogens and detrimental microbes in our bodies.
Oleh karena itu, perusahaan kami membuat minuman probiotik nanospray yang dispraykan pada kulit dan hidung. Kami sedang membuat penelitiannya. Kemudian membuat juga air microbiome, ya, probiotik yang dispraykan ke ruangan. Kami juga memproduksi probiotik untuk hewan ternak.
And because of that, our company has taken a step in which we have developed products that will support the microbiome of the air in the environment through a probiotic humidifier, And we also have came up with nano sprays that can be applied to our noses and mouths, to our digestive and respiratory tract. And we have also figured out probiotics that can be used for livestock.
Penting sekali fungsi mikrobiota memodulasi fisiologis tubuh ya, termasuk modulasi hormonal, modulasi imun sistem, sehingga kita bisa sehat terhindar dari berbagai macam penyakit, mulai dari penyakit infeksi dan penyakit kronis.
And by taking probiotics, we'll be able to modulate our body's physiology and the way it works, so that we can be more immune to ailments and diseases and have a more balanced health.
Tapi sayangnya probiotik yang merupakan komponen utama dari mikrobiota, minimum 80%, mudah gugur ya, mudah mati, sehingga kita perlu suplementasi setiap hari, daily supplementation.
Unfortunately, in order to attain about 80% of the recommended composition of probiotics in our gut microbiome, there are challenges in which our beneficial microbes and probiotics in our bodies might collapse or even get wiped out easily, which prompts for the daily consumption of probiotics.
Pertanyaan saya, untuk membangun mikrobiota yang beragam dan seimbang, atau merestorasi mikrobiota yang rusak, disbiosis, diperlukan probiotik yang tunggal atau probiotik multistrain. Karena probiotik tunggal disuplementasi tiap hari beresiko terjadinya ketidakseimbangan, ya, change the balance of microbiota.
And so my question is, in order to maintain the diversity and balance of the gut microbiome and the body's microbiota, do you think it is wiser to consume daily single-strain or multi-strain probiotics for daily supplementation? It's because single-strain probiotics might have a chance of tipping the composition of the gut microbiome to one side, which will eventually risk the imbalance of the gut microbe composition.
Thank you.
Thank you. Thank you. Very interesting question. Very interesting. Your company has products, different kinds of products, including nano products, yeah? And for sprays, and your son speaks very good English, very American accent. Yeah? So you ask $1 million question. Yeah? I do not think I can answer that. Single strain or multi-strain? I think many people want an answer to that question. I do not think I can answer that question. And there could be, we can argue for another one whole day, another webinar on that. Maybe Professor Endang, Professor Ingrid are more qualified to speak about that. Yeah? So I cannot answer about that. But it's very interesting that your company has shown interest in this and do your own R&D, your own product innovation and product R&D, and come up with new ways of getting probiotics to the people, which very short-lived. Yeah, we must get to the people every day. So maybe other experts in probiotics can discuss about your question: single strain or multi-strain? Thank you, Madam Chair.
We appreciate the answer. Thank you so much.
Yeah.
Thank you, Bapak G. Rektas Gesen. Would you like to invite Professor Endang to try to answer the question from
Professor Endang?
Bapak G?
Professor Endang, you are our guru in the C probiotic strain. Professor Ingrid.
Ya, terima kasih, Dr. T. Bu Moderator, can I explain about the single strain, multi-strain? Yeah. It's everything should be based on scientific evidence. Even single strain, if powerful, then we can say that Single strain also have a health benefit. Multi-strain, if the microorganisms cannot be identified clearly, we cannot distinguish actually which one give more powerful than others. So, yeah, I'm a researcher. Yeah, I'm researcher. So that's why to prove that multi-strain it's better than single strain, it's actually not easy. We must really read the result, the papers, yeah, from the publication. Yeah, that's we can what you call it? Yeah, we can believe. Yeah, the benefit of the probiotic strain. Probiotic strain, the benefit of probiotic strain should be explained or should be published in paper. Yeah,
that's true. So
yeah.
if I
scientific paper. So
that's right.
The regulators at the BPOM only what to say? Only read from the paper.
Yes,
yeah,
they
the
want proof.
evidence.
Yeah.
This strain it's really give benefit.
Yeah.
The paper must be the research must be done by the what's we call it RCT, RCT double blind.
Yes.
Yeah, okay. Yeah, it's common. It's common. So to prove that the strain it's really probiotic, it's long way to go.
That's true. And you're right, Prof. Endang. There are many papers which publish on single strain demonstrated beneficial effects also. And of course, there are also multiple strain. I also mentioned just now, if there's multiple strain, they must prove that they work together. They don't fight each other.
Right. That's must be proved by publication. So we can only read from the papers. Yeah.
Thank you.
Yeah.
All right. Thank you very much, Prof. Endang, for your comprehensive explanation and also combined from Dr. T. Thank you. I would like to go to the last panelist for this session. I would like to invite Director for Traditional Medicine and Health Supplements Control, Bapak Irwan SSIAPT MKM. Please, the floor is yours.
Good afternoon, Dr. E. Siangti and Professor Seppo Salmian. I am Irwan from Badan POM, Indonesia FDA. Maybe I'm not the last panelist of this session, but last panelist of this seminar, right?
Thank you for correcting me.
Your board presentation is impressing me and also enrich our knowledge regarding regulation on probiotic and its implementation. We hope we can conduct supervision on probiotic and more effective than before. Our concern is how to make probiotic as health supplement, maybe not related to claim of probiotic. I have two questions. The first question is, we know probiotic are made from living creature, microbe, that very different characteristic than ingredient that commonly used in health supplement, such as chemical substance on herbal. Therefore, maybe we need special handling during the manufacturing process. In context of manufacturing standard, maybe good manufacturing practices or HACCP, is there any specific manufacturing requirement for probiotic as health supplement? Is a first question. And the second question is, in order to maintain the purity of probiotic species and strength, should the fermentation of probiotic be carried out in a sterile environment? If the probiotic is fermented in non-sterile environment, is there any impact of its quality? There are two of my question, and we hope that we can have explanation of yours. Thank you.
Thank you, Dr. Irwan. And I must congratulate your division for organizing this seminar today. I think you have touched on many aspects, including research and development, regulations, requirement for science. So I think, and you have also multiple stakeholders together in this platform to share the discussion on experiences, on challenges and opportunities. I think this kind of openness in Badan POM is very good, and Badan POM has been supporting the Southeast Asia probiotic scientific and regulatory experts framework for a long time, even starting from the beginning. Yeah, and so regarding your question on the hazards, HACCP requirement, yeah, you're right. Probiotic microorganism is totally different from other components you put into a food, vitamins or minerals or other ingredients. So the requirements must be very, very different for the probiotic. And one can never imagine, let's say 30 years ago, that probiotics, microorganism can become beneficial to health. Yeah, people think of microorganism, bacteria as bad, as harmful to health. So we have to spend some time to explain to the public that bacteria can also be good bacteria and also bad bacteria. Unfortunately, I'm not in the area of food safety, and I am not able to explain about hazards or HACCP or good manufacturing practice. And also your second question about sterile environment. Yeah? So there are several industry people in this room. Maybe they can help to share their thoughts in this. So I must apologize. I am not expert in this area of food safety or manufacturing practice. So very often we also learn from the food industry what are the manufacturing practices that they learn from their experiences, what are useful practices, and share their practical experience with academic people, regulatory agencies. Maaf ya, Bapak. Ya.
Thank you, Doctor.
All right, so all the two questions already answered by Dr. T, right? All right, thank you. So I think that will become the end of this session. I would like to thank the speaker, Dr. T, for your informative, and also all the panelists today. We have Prof. Endang, we have Bapak Slamet, and we also have Bapak G, and also Bapak Irwan. For the panelists, thank you very much for the productive discussion. And I think, yes, this marks the end of discussion. Thank you very much for your attention. I will give back to MC. Thank you.
Thank you very much for the speakers, panelists, and moderator. The distinguished ladies and gentlemen, we are now at the end of the meeting. Alhamdulillah, we were able to complete this seminar in such a successful and fruitful manner. Now we would like to commence the closing ceremony for this virtual seminar and exhibition under the theme Research and Potential Development of Probiotic Products for Health and Regulatory Study of Probiotic in Other Countries. Hence, I would like to kindly invite Ibu Reri Indriani, Deputy for Traditional Medicine, Health Supplement and Cosmetic Control, the Indonesian Food Drug Authority, to deliver the closing remarks and cordially close this virtual seminar and exhibition.
Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon, distinguished ladies and gentlemen, and also all experts and the panelists from the industry, health associations, and also from the manufacturer or importer association, and also from the representative from the consumer or the community. First of all, I would like to thank to all participants of this international scientific seminar. And of course, we have to say Alhamdulillah, but that the international scientific seminar has been conducted successfully and will provide benefits for all of us. In general, what have been delivered by all the speakers, all the experts, and also input from all the participants of the seminar, we can conclude some important or some important points, such as that we do agree that the probiotic is a living microorganism that when administered in adequate amounts in a health will provide a health benefit on the host, as long it proves or fulfill the safety, efficacy, and quality, referring to the established guideline and supported by scientific data. And we have known that so far, the use of antibiotic, the approved claim, particularly in Indonesia, in ASEAN, that we have heard from the speakers, is have limitation only for the gastrointestinal health, to support the gastrointestinal health. But realizing that there is still potential challenge to broaden the claim, to explore the benefit, Indonesia, in this regard, Indonesia FDA will take the opportunity to overview, to study other country regulation, standard, or best practice in order to increase the access of community to have broaden usage of probiotic. Through the deep and comprehensive discussion during the seminar, we also know that there are still some obstacles in providing the scientific data to support the safety efficacy of the probiotic regarding the new strain or the new claim. Even though that WHO, and FAO has stipulated guideline on probiotic in 2001, which been used as the reference by others country, by so many countries. Any information, regulation, standard, and best practice will be used by Indonesian FDA to study, to overview further, in order that Indonesia FDA will provide the updated guideline so that in the near future, Indonesia will have the new guideline in evaluating probiotic product, while on the other hand, also to increase access for the community to have probiotic which have a broadened claim. I think that's all from me. With that statement, I finish my closing statement, and thank you very much for all participants who have been called this seminar since 8 o'clock this morning till the last session now at 3 o'clock in the afternoon. Thank you very much. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Ibu Reri Indriani. With this, all the programs of virtual seminar and exhibition, research and potential development of probiotic products for health and regulatory study of probiotic in other countries has been completed. Kindly, all participants can fill the evaluation form. The link evaluation form can be read in the chat box. This evaluation is needed for us to know and enhance the quality of the seminar. We would like to thank you for your participation during the seminar, and as the host, we apologize for any inconvenience during this seminar. We wish you all very pleasant day, stay safe and healthy. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Diskusi-diskusi yang lain menyambung ini. Tadi cerita banyak juga formularium fitofarmaka.
Paling manuk cingklang cing datang, loskan kolom bapak satan bulanan. Mendaki jalan pandai sikit, menurun jalan kebiaro, di hati indah mampe awak kaki cuak, oi oi ayam dan lape. Layang-layang, selayang pandang, hati di dalam, bagai bergoncang. Jangan ragu dan jangan bimbang, ini lagu selayang pandang. Gandang garam tu, hayat dengan kangkalu, sambil tuntang gang tahu, niat mundur dan maju. Ayo menari manasai, menari manasai, menari manasai. Belenggang patah-patah, nganape goyang pica-pica, nganape body bocor-bocor. Tenggang-tenggang lopi na anak koda ipanjajah buluana. Pinang muda dibelah dua, gunung kerinci, gunung yang tinggi menjulang. Wahai anak muda, kalau berjalan harus berdua, senanglah hati walau tiada orang yang larang. Mana di mana anak kambing sayang, anak kambing tuan ada di pohon waru. Mana di mana jantung hati sayang, jantung hati tuan ada di kampung baru. Caca marica hei hei, caca marica hei hei, caca marica ada di kampung baru. Caca marica hei hei, caca marica hei hei, caca marica ada di kampung baru. Ayo mama, jangan mama marah beta, dia cuma pegang